our patron, Dr. Sister Jyoti, Principal Nirmala College, and our honorable resource person, Professor Emeritus Pathophysiology Mazumdar, have joined. So with the permission of our patron, I will start this session. Respected Dr. Sister Jyoti, Principal Nirmala College, Ranchi, respected resource person, Professor Pathophysiology Mazumdar, Distinguished Professor and Founder, National Institute of Biomedical Genomics, Co-Convener Dr. Anubhuti Singh, and my dear participants, a very warm good morning to you all. On behalf of the Departments of Botany and Zoology, Nirmala College, Ranchi, I welcome you all to this 2 days national webinar on pandemic COVID-19, pathophysiology, prevention and treatment. Dear participants, since its emergence in Asia last year, the coronavirus Sars Cov-2 has spread to every continent. There is no doubt that it has the potential to adapt. This evolutionary changes has worried us, as it can lead to changes in transmission, in the duration or severity of disease. We have with us Honorable Resource Person, Distinguished Professor and Founder, National Institute of Biomedical Genomics, Professor Pathophysiology Mazumdar in session 1, who will be presenting on this burning issue under the title, Going Viral, Evolution and Spread of SARS-CoV-2. Let me speak a few words for Sir. Professor Pathophysiology Mazumdar is the founder of National Institute of Biomedical Genomics and concurrently is a professor of Indian Statistical Institute, Kolkata. Sir, your scientific contribution to human and population genetics span a vast range from structure and evolution of human population to genomics of disease and development of statistical methods for genomic analysis. You are Sir J.C. Bose National Fellow and an elected Fellow of the 3 Science Academies of India, of the World Academy of Science and the International Statistical Institute. You are the Council Member of Human Genome Organization. You are the Indian National Coordinator on the International Cancer Genome Consortium. You have served on the Board of Directors of International Genetic Epidemiology Society and was the founding chair of its ethical committee. You have worked for the International Bioethical Committee of UNESCO. There are many awards to your name. Sir, we are really honored to have you as our esteemed resource person. I request you to begin this session.
Thank you very much. And first of all, let me wish all of you a very good day and a very safe and healthy months to come. We are going to talk about a pandemic and about the virus that has actually caused this pandemic. In our lifetimes, those of us who are present in the audience, including myself, I don't think we have ever faced a pandemic. So this is for the first time that we actually face a pandemic. The last pandemic was in 1918, which was named as the Spanish flu, and it killed millions of people. This virus is not killing as many, but is transmitting and infecting many people. And as was just told, that it is not restricted to one geographical region, it's spread across all continents. We are going to talk about this virus, so I hope that you will stay safe and be healthy. Like I said, that I'm going to talk about this coronavirus, but let me first begin by saying make some general comments about viruses in general. So these viruses are very funny. They are neither living entities nor non-living entities. They are in some ways very funny. They are non-living primarily because they cannot reproduce, they cannot replicate. These viruses usually are naked nucleotide acid sequences, DNA or RNA that's wrapped in a protein coat. This particular virus, the coronavirus, the SARS-CoV-2 coronavirus, has a lipid coat, and that's the reason why we are all asked to wash our hands with soap every so often, because if we wash our hands with soap, it sort of disintegrates the lipids and exposes the naked nucleic acid sequence, which in this particular case is an RNA sequence. The coronavirus SARS-CoV-2 is an RNA virus. It exposes the RNA, and an exposed RNA doesn't last long. So it essentially kills the virus by washing our hands with soap or sanitizing our hands with alcohol and other such chemicals. This coronavirus is an RNA virus, like I just pointed out, and is the longest known or the largest known coronavirus, largest coronavirus that we know. It has 30,000 alphabets in its RNA nucleotide sequence, and it is the longest RNA virus. It also has other kinds of properties. It's positively stranded, and it's positively stranded. That means that it is already prepared to make proteins, but it cannot make proteins by itself, or at least cannot make those proteins that enables it to replicate. It has to get into a host cell, so it's an obligate parasite. All viruses are obligate parasites. This particular virus is also an obligate parasite. It infects human cells, but not just human cells, also other cells of other organisms, and I'll come to that in a few minutes. This virus has been causing panic all over the world, has spread all over the world. As all of us know, it began in Wuhan, in a seafood market. A lady who was handling seafood came down with fever, cough, and other such symptoms that we are now familiar with. At that time, the doctors there in Wuhan were not familiar, but then she was coughing, and they believed that there was a lung infection. Essentially, then they isolated the coronavirus from the lung, and it turned out to be a virus and a coronavirus at that. The lady recovered, and she's still living. This prompted people to start identifying or start asking the question, where did it come from? This is the first time that we are meeting this particular kind of coronavirus. Where did it come from? So that led to some kinds of evolutionary questions. Where did it come from? And one of the ways to answer these kinds of questions is to sequence the RNA. So sequence the nucleotide. So in this particular case, it was the RNA that was to be sequenced. So they sequenced this RNA virus, they sequenced the RNA nucleotide sequence that was within this particular coronavirus that was isolated from this woman. And they then looked at this sequence in relation to other sequences that were already known. So there is a database of these RNA sequences of other viruses that are known from the humans. There are many viruses that infect the humans. Other viruses. There are also other viruses that infect other organisms, and in particular, the bat is full of viruses. So the bat is home to lots of viruses, and it's been estimated that the bat is home to about 67, maybe 70 different kinds of viruses. Why the bat has to be such a wonderful home for so many different kinds of viruses is not completely known. There are some speculations as to why the bat harbors so many different kinds of viruses, but I will not get into what those speculations are. So they started comparing people who were scientists who were interested in finding out where this coronavirus came from. They started investigating and comparing sequences. And one of the ways that we try and identify these kinds of evolutionary aspects is to compare sequences. And the framework is that if we identify another sequence that is very similar to the sequence that is in question, then we would say that that particular sequence is the ancestor or a relative of this particular sequence. So when they looked at this database of RNA, of sequences of viruses, of sequences of RNA viruses, lo and behold, what they found was that this sequence was very, very close to a coronavirus that was found in the bat. Now, what do we mean by close? So when we compare two nucleotide sequences and we kind of align them, then we look at alphabet by alphabet and ask ourselves that how many of these alphabets are they exactly the same? Do they match? And with respect to the coronavirus that was found in the bat, and with respect to, and this particular human coronavirus, SARS-CoV-2, the nucleotide match, the exact identity between these two nucleotide sequences is 97%, which means that if you take a random sequence, subsequence of 100 nucleotides from each of these two coronaviruses, the bat coronavirus and the human coronavirus, on an average they will be exactly same nucleotides at 97 of these 100 positions. So they are very, very similar, and so people inferred that this particular coronavirus must have crossed over from the bat to the human. Now, this interspecies crossing over, called the zoonotic crossover, happens, and it happens because of certain kinds of DNA or RNA changes that take place in the viruses that gives it the ability to make this crossover. And these were identified, and so it was quite clear that there was a zoonotic crossover from the bat to the human. Next question is, it's known that they don't oftentimes cross over directly. There is an intermediate host to which they come, and then from that intermediate host, it goes to the human. The question was asked, could there have been an intermediate host? And it was kind of by accident an intermediate host was found. We are still not 100% sure that that was the intermediate host. The way that it was found is that about 150 kilometers away from Wuhan, somebody found accidentally two anteaters, and these anteaters are called pangolins. It's a scaly anteater, two anteaters who were dead, and it was evident that they had some problem because there was a lot of froth coming out of the mouth. When a lot of froth comes out of the mouth, the standard suspect is that most likely there has been an infection in the lung. So they autopsied these two pangolins, and they looked at these. They looked at the lungs of these pangolins, and they found the virus. And it turned out to be a coronavirus that was related to the bat coronavirus. And so it's been. And they also compared it to the human coronavirus. So now we think that the way that it has come to humans is that this coronavirus had its home in the bat. It transmitted. There was a zoonotic crossover from the bats to the pangolin, and from the pangolin to the human. So do we come very close to the pangolin? We probably come closer to the bats because of lots of trees that are being felled, etc., and buildings that are being constructed. These homes, these new constructed buildings, when they're unfinished, you will see that there are lots of bats hanging from the ceilings and so on. So they provide an intermediate shelter because the trees don't exist. So we've come in much closer contact with the bats. They would be in the forest, but right now there are no forests, so we've come in closer contact with the bats. But do we come in close contact with the pangolin? Usually not. But what had happened is that most likely, and this is reconstruction, most likely this is what had happened is that in this particular seafood market in Wuhan, where the first outbreak took place, they also sell pangolin meat, and so this there was a lot of handling of pangolins in that particular seafood market. Although pangolin is not seafood, but along with seafood, they also sell other kinds of exotic meat, and so it's likely that this particular pangolin that was being handled for selling meat actually carried this coronavirus, and that's how it transmitted to the human. So the pangolin part of this story is not very clear, but the bat part of this story is without any controversy at all. Some people believe that it may not have been the pangolin, but again, the jury is out. It will be settled quite soon. So this is how we believe that we have gotten the coronavirus SARS-CoV-2. Are there other coronaviruses that are known to have infected humans? Indeed, there are 7 different coronaviruses that are known to have infected humans. Two of them I will narrate to you. The remaining 4, SARS-CoV-2 included. So SARS-CoV-2 and 2 others, the 2 others I will narrate to you in a minute. There are 4 others that also infect humans, but they don't cause as much panic in humans because they don't precipitate as much disease. They cause mild flu-like symptoms, and they are with us. They remain with us, but they don't bother us so much because they don't kill us, and they only cause mild symptoms in us. The other 2 viruses that I wanted to talk to you about is, again, another virus, which was a coronavirus that was named SARS-CoV, and that was also, that originated in the human, in Wuhan, as a matter of fact, in 2003. And that particular outbreak essentially died a natural death. It didn't become an epidemic, and certainly did not become a pandemic. It was named Sards Coronavirus, and SARS, the name SARS is because this particular coronavirus goes and attacks the lungs and creates a respiratory distress. So SARS stands for Severe Acute Respiratory Syndrome. Syndrome means that it just not only causes respiratory distress, it causes other kinds of problems, one among them being diarrhea. And I'll come back and tell you in a minute why it also causes diarrhea. We now understand why it causes diarrhea. So it's a syndrome. It's a plethora of various kinds of clinical conditions that it causes, but primarily it's an acute, severe acute respiratory distress that it causes. So this coronavirus, SARS-CoV, was contained. It caused some amount of respiratory distress in some individuals, but it was contained, and we didn't believe that there would be another relative of the SARS-CoV that would again raise its head 20 years later. So almost 20 years later, we have another, 17 years later, we have another coronavirus, which is related to the first coronavirus, SARS-CoV, that should then have been named as SARS-CoV-1. Now it's called SARS-CoV-2, the present coronavirus. So that outbreak took place in Wuhan. It was contained. It didn't create too much of a problem. The next coronavirus outbreak was in the Middle East, and that Middle East coronavirus is called the MERS coronavirus, Middle East Respiratory Syndrome coronavirus, MERS. That had also an intermediate host in the camel, and so on and so forth. Let's not get into that. That MERS coronavirus was kind of dangerous. So let me now tell you which is among these 3 viruses is the most dangerous. Which of these 3 coronaviruses is the most dangerous? Let me provide you with some data. The remaining 4 coronaviruses are not dangerous at all because they don't actually create fatalities. They don't cause death. They only cause mild flu-like symptoms. So if we compare these 3 coronaviruses, the current coronavirus, SARS-CoV-2, is the most kind of these 3 coronaviruses. So it is a very kind coronavirus. And why do I say that? Because it is not killing people at all. So let me give you some comparative figures. Of the 100 people that the SARS-CoV in 2003 that it infected, of the 100 people that SARS-CoV infected, 11 persons on average died. So 11% was the fatality rate. 11% was the rate of death among infected individuals. When the MERS outbreak took place, that was in the Middle East, and that took place in 2012-13, 10 years after the first SARS-CoV outbreak, the number of people that particular coronavirus killed was 34%. So of the 100 people that it infected, 34 people died. So 11%, 34%, and now this particular coronavirus is probably killing less than 1% of the people that it infects. There is, of course, a huge amount of death, mortality among the elderly, people who are above 65 years of age. If they get infected, the death rate is much more, but across the board, across all ages, the overall death rate is less than 1% or just about 1%. So 11%, 34%, 1%. So I have actually written in popular science articles calling this coronavirus as a very kind virus. This particular coronavirus is kind. It doesn't kill many people, but it spreads very well. And we know that it spread across all continents. After the 1918 pandemic, the next pandemic to hit humankind is in 2020. over 100 years later. So this particular pandemic, this particular coronavirus spreads itself very well. So it spreads good. Now I will come to talk about two things. One is how does one track spread, but I'll come to that a little later. Let me tell you about what the virus does in order to infect a human. I have already told you that viruses are obligate parasites. They cannot reproduce by themselves. They have to get into a host cell, and in this particular case, humans are hosts. They have to get into a host cell and hijack the host cell machinery to their advantage in order to reproduce. So this is what all viruses do. In particular, this coronavirus preferentially infects the lung cell. So when the virus, which is again, like I said, a naked piece of DNA or RNA wrapped in a protein coat, comes in contact with the human, we breathe in the virus particle, and usually the virus particle is encased in a droplet, and these are droplets that when we speak, we are constantly producing these droplets, and the viruses are encased in these droplets. We inhale them from another person. When another person is speaking and producing droplets, if that person is infected, many of those droplets will contain the virus, and when we inhale the virus, they actually go to our lungs. And what do they do after going there? How do they get into the lung cell? So the first thing that a virus does is to go and attach itself to the surface of the lung, to the surface of the cell, in this particular case, the lung cell. After it attaches itself to the surface of the cell, it must actually crack open the cell membrane of the host and get into the cytoplasm of the cell. So this is what this virus also does. It cracks open the cell membrane of the lung cell, and then gets into the lung cell, and then starts hijacking the host cell machinery and starts replicating. And once it starts replicating, as you're talking, as you're coughing, you're giving away these droplets that encase viruses. Somebody else is breathing in, and therefore that somebody else is also becomes infected. So the virus is able to spread, and that's Darwinian natural selection. So the Darwinian natural selection plays in this fashion, that the virus goes and attaches itself to the host cell. It enters the host cell. Once it enters, it starts hijacking the host cell machinery, starts producing appropriate proteins for replication, and then it spreads to other individuals. So the virus is then able to make many more copies of itself, and that's Darwinian natural selection, positive natural selection. So a corollary of this is that if the virus kills the host, if the virus somehow damages the host lung cells and kills the host, then the virus does not get a chance to propagate itself. And that's not to the advantage of the virus. The virus does want to propagate itself. So a virus that is less kind does not propagate so well. So the MERS virus, which killed 34 of the 100 people it infected, did not propagate very well and therefore died a natural death. The same thing happened with Sars-Cov, but this one is killing much less and therefore is able to spread better. A little bit of biology of how it attaches itself to the host cell and what it does in order to enter the host cell. So this particular virus is called the coronavirus, and the coronavirus, as you know, it looks like the flares of the sun. You've seen pictures of it. It's like a round ball with lots of spikes. These are called spike proteins. The spike protein actually helps this virus to anchor to the host cell. So the spike protein attaches itself to another protein that is on the cell surface, that's on the surface of the cell of the humans, and it attaches itself. Now we know the spike protein of the virus attaches itself to another protein that is found in plenty on a specific kind of lung cell called type 2 pneumocyte. Type 2 pneumocyte is a special kind of lung cell. Lung cells are very heterogeneous, but type 2 pneumocytes exhibit or produce a specific kind of protein on its cell surface, and that specific kind of protein is called ACE2, angiotensin converting enzyme 2. Doesn't really matter in terms of the concept, you don't need to remember the name, but it expresses a certain kind of protein on the cell surface to which the spike protein of the virus goes and binds, goes and anchors. So that's how the virus is able to anchor itself, find its temporary home on the surface of a cell. Then it needs to crack the protein and crack the cell membrane and get inside of the host cell and into the cytoplasm. I'll tell you that part of the biology of that part of the story in a minute. But I talked to you about ACE2 protein, to which the spike protein goes and anchors itself. To the type 2 pneumocytes, are the type 2 pneumocytes the only kind of proteins only kind of cells that expresses on its surface the ACE2 protein? The answer is no. There are many other cells in the human body that also express type 2 that also express ACE2, other than the type 2 pneumocyte that also expresses ACE2, other cell types. And one of the cell types is the enteric epithelial cells. The enteric epithelial cells are full of the gut and the colon and so on and so forth. Those cells also produce a lot of ACE2 protein on its surface. So if the virus is able to get into your body, it also goes and finds other kinds of cell types that express ACE2. And one of the cell types that expresses ACE2 is the enteric epithelial cells, which essentially means that it goes and creates problems in your gut and colon, in your gut primarily, and it gives you diarrhea. That's why we find that many people infected with SARS-CoV-2 shows clinical symptoms of diarrhea. Many people, I'm told that about 30% of the people have severe diarrhea if they are infected with SARS-CoV-2. Aside from, of course, the major clinical symptom is respiratory syndrome and respiratory distress. Okay, so now we've understood how the SARS-CoV-2 goes and anchors itself to a specific kind of cell that expresses to specific kinds of cells that express ACE2 protein. In particular, we will talk about the lung cell. Now, once it goes and anchors itself, the spike protein also has to use, again, I'm trying to simplify the biology. So the protein has essentially two components, and these two components are named as S1 and S2. And there is a point between, or a region between S1 and S2, which needs to be cracked in order for the protein to become active, for the viral protein to become active, that will actually be able to fuse the viral cell membrane, and the host cell membrane. That particular cleavage, technically it's called cleavage. So the S protein comprises two components, S1 and S2, and there is a cleavage site in the middle. And this cleavage is done by the host protein, very interestingly. It's done by a host protein, and this particular protein is serine protease. Proteases are proteins that chews up other kinds of proteins. And so this S1, S2 cleavage is done by this particular protease that will go and chew up that particular region and make the two components S1 and S2 extremely active, and that activity in turn allows the virus to actually crack open the host cell membrane and get into the host cell cytoplasm, after which it does its usual activity, which is replicate itself and make other kinds of proteins as well. So this TMPRSS2, the protein serine protease, is a host cell phenomenon and is a host cell protein that helps the virus to enter its own cell. So this is essentially the biology. The biology has two components. One is that the virus will need to go and attach itself to the host cell membrane, and then it uses the host cell protein to crack open the cell surface of the host and enter the cytoplasm, and then starts its action from there. And then it starts behaving like a living being, like a living organism, and it replicates itself and it spreads itself. So that's the part of the biology of the story. There are other complications, there are other nuances to this particular story. I'm not going to get into the detail. The next part that I actually want to mention to you are two interesting things that happen. Well, one is with respect to tracking. So we say that this virus is spreading. How do we know that this virus is spreading? Is there an objective way to know when that this virus is spreading? And secondly, can we actually track the spread of the virus and decide for ourselves that the spread is being contained, it's coming down, and therefore the COVID-19 disease will not spread in the population anymore, because the virus is, the prevalence of the virus, the spread of the virus is coming down. So we need objective measures. Let me tell you, so these objective measures are statistical measures, but I'm not going to get into the statistical details of it, but let me give you the concept as to how we track and how we get to know whether the virus is actually reducing its spread. So when a virus wants to spread from an infected individual, it needs to go and infect another uninfected individual. Once you get an infection, once you're infected, you start producing your own immunological proteins. Your immunological machinery then becomes active, and the virus, once it enters an individual who has these appropriate immunological mechanisms that are active, will not allow the entry of the virus. So an individual who is infected once usually doesn't get reinfected again in a reasonable period of time. We don't know what that reasonable period of time for this particular virus is, but there are viruses that provide us lifelong immunity once we are infected. There are viruses that provide us with 3 or 5 years of immunity, and so on and so forth. I won't get into the detail, but essentially, for a reasonably extended period of time, we do not usually get reinfected if you've been infected once. The same thing is expected of the coronavirus as well. So the virus then needs to find an uninfected person, a person who's never been infected with this coronavirus, in order to infect and spread itself. Once a virus is unable to find another uninfected person to infect, then the virus is, obviously the virus is not going to be able to spread, because it spreads by infecting uninfected people. So the way that we track is, we ask ourselves, on an average, one infected person is able to infect how many uninfected people? If that number is greater than 1, that means one infected person is infecting more than one uninfected person, which means that the virus is spreading. More and more people are getting infected. When that particular value becomes equal to 1, one uninfected person is able to infect one uninfected person. One infected person is able to infect one uninfected person. Then it's stabilized. Then it's a plateau. Once that value gets less than 1, so let's say that on an average, one infected person is only able to infect 0.5 uninfected people. That means 2 infected persons are able to infect only 1 uninfected person. Then we know that the prevalence of the disease, the spread of the disease, is going to come down. This particular number you may have heard, and so let me tell you this number. It's a value that's called R0. So R0 is the average number of people that an infected person can infect. The average number of uninfected persons that an infected person can infect. If it is greater than 1, it is spreading in the population. And when it becomes equal to 1, it's plateaued, it's become constant, and when it gets less than 1, we know that we've controlled the disease and it's coming down. How do you actually control R0? One way to control R0 would be, for example, if there are many, many people in the population who have become infected and therefore have become naturally immunized to the virus. So if the virus spreads in the population, it will infect many people, but after a certain period of time, it will not find many people to infect, and therefore it's going to die a natural death. We can ask ourselves, what should be, if you have a large population, what is the proportion of the population that must become immune to the virus? In other words, must produce certain kinds of immunological proteins in the body that will not allow the virus to infect this person. And like I said, that you can either be naturally infected by the virus and recover, in which case you will not be reinfected, in which case you will go out of the susceptible pool of persons whom an infected person can infect. So what must be the total number of people who should be become immune in order for the virus to actually die a natural death? We can estimate this number under certain assumptions, and that number turns out to be 67% of the population. Varies between 60 and 70% of the population must become immune to must produce immunological proteins in the body against this particular virus, for this virus to die a natural death. It takes a long time for 60 to 70% of the population to become naturally immune. So how else can we become immune? Another way that we can gain immunity is by a vaccine. A vaccine is like an artificial virus. It's a surrogate of the virus. It produces, it's artificially injected or orally taken, whatever, ingested in the body. And once that is done, that particular virus or that particular vaccine will mimic the original virus, and it will evoke a similar kind of immunological response in the individual who is being vaccinated or who's just been vaccinated. In which case, you gain immunity not by a natural infection, but by artificial immunization or artificial vaccination. So we are all waiting for this virus to come so that those of us who have not yet been infected can take the vaccine and become immunologically active against this particular virus. And therefore, we hope that we will not be infected, and then we will go out of the susceptible pool of people whom the virus can actually infect. So these are two ways, and that's the reason why there is so much of rush and push to find a new vaccine for this. Where do we stand with respect to our vaccination efforts? With respect to our vaccination efforts, as you know, that lots and lots of people are trying to find vaccines. And there are many, both companies and researchers who are actually looking for vaccines against this particular virus. And there are two vaccines that are advanced, at an advanced stage. India also has two vaccines that are relatively advanced, but not as advanced as the other two vaccines. The two vaccines that I'm talking about, again, you've read in the newspapers, maybe you are a virologist, so you know the details. There are two vaccines that are really at an advanced stage. One is the Oxford vaccine, which is Oxford in collaboration with a pharma company called AstraZeneca. So AstraZeneca Oxford vaccine is one that's advanced, and I'll define what I mean by advanced. And the other vaccine is, Oxford is in the UK, AstraZeneca is a global pharma company, but they have tied up with the UK enterprise or UK organization. So let's call that as the UK vaccine. The other vaccine is the US vaccine that is being that's thought through or manufactured by a company by name Moderna, and that particular vaccine is also fairly advanced. In India, we have two other vaccines, one's Cadila and the other is Bharat Biotech. The Bharat Biotech vaccine is in collaboration with, well, both of these are actually in collaboration with scientific institutions, but one is being pushed forward by a pharma company called Bharat Biotech. The other is being pushed forward by a pharma company called Cadila. They are not as advanced, not at as advanced a stage as the other two vaccines. So what do I mean by being at an advanced stage? The major thing that I wish to tell you is that in the process of vaccine discovery, there are three things that we need to do. First of all, we need to get hold of a vaccine, get a vaccine construct, as they are called. And then do three things. One is test the safety of this particular vaccine. If we inject this vaccine or feed this vaccine to somebody, will that person come out with very great amount of huge adverse events, will fall sick in a major way. So we need to identify, and if anybody falls sick, then of course we can't carry that vaccine forward. That's called phase 2 trial. It's usually done on a small number of individuals, maybe 50 or 100 individuals. who volunteered to use that vaccine and hope that they will not fall sick. With respect to both the Moderna and the UK vaccine, and also the Indian vaccines, the Cadila and the Biotech vaccine, there were no reported major adverse events. It passed through the phase 1 trial, which is the safety trial. Moderna vaccine, I think in one individual there was a problem, but I think I don't know the details of that, but there was one individual showed some adverse events of the number of individuals that they had actually injected with the vaccine. The second part is to test whether the vaccine is efficacious, meaning that does it actually protect you against the coronavirus. So that is done in a slightly larger number of individuals. Once the vaccine is proven to be safe, you want to find out whether the vaccine is effective. So that's called phase 2 trial. Then you go through another set of individuals that you inject, and that's called the phase 2 trial. All 4 of these vaccines that I'm talking about have gone through the phase 2 trials without any problem. They have proven their efficacy. They are all not at the same level of efficacy, but acceptable levels of efficacy. The third is the more difficult trial, which is called the phase 3 trial, where the number of people on whom the vaccine must be used should run into tens of thousands. So the Oxford UK vaccine, and they're all volunteers. You can't force anybody to use the vaccine. So these are all volunteers. The Oxford vaccine has roped in about 30,000 volunteers. So 30,000 people said that they will either take the vaccine or, so they will participate in this trial that comprises the phase 3 trial of the vaccine. And what do we try to ascertain from the phase 3 trial using such a large number of individuals? Essentially, whether it gives adequate protection. The phase 2 trial has provided an indication, but that was based on a small sample size. So now we want a much larger sample size. We want them to go about doing their daily chores and then estimate what proportion of the people who have taken the vaccine have fallen ill. So if that proportion is significantly lower than the proportion of people who have fallen ill but had not taken the vaccine, then we know that the vaccine is working and the vaccine will protect people against the coronavirus. The Oxford and the Moderna are just starting their, or have just started their phase 3 trial. The Bharat Biotech and the Cadila, they are getting prepared to start their phase 3 trial. They're still recruiting volunteers to start their phase 3 trial. So as you can imagine that we have to wait for a sufficient number of time, sufficient period of time for people to fall ill, people who have not taken the vaccine to fall ill, etc. There are some ways of, speeding up this process. I will not get into those technical details of speeding up this process. So they are trying to speed up this process by a specific study design of the phase 3 clinical trial. Having said all of this, we don't know. We still have to wait. Again, nobody has an estimate of the time, but it's estimated that we may not actually get the vaccine within the year. Once it passes the phase 3 trials, then you need to actually manufacture the vaccine. The Oxford group has been very proactive, and they have contacted a large number of manufacturers all across the world, including our own Serum Institute of India, which is the world's greatest producer of vaccines. They don't invent vaccines, they produce vaccines, or maybe they invent a few, but they are known the world over as being the manufacturers of vaccines, and they are the world's largest. So the Oxford group has already tied up with the Serum Institute. If their phase 3 trial goes through, they will actually have Serum Institute as one of the components who will produce a large number of doses of the vaccine. So this is where we stand with respect to the vaccine. And the one other last point that I will tell you, and I won't tell you in any detail, is that this particular virus, as we all know, has spread throughout the world. What has happened, as a matter of fact, in an interesting sense, that after it arose in Wuhan, it spread to other parts of the world, and as a virus spreads to other parts of the world, it evolves. And what do we mean by evolution of a virus? What it means is that in the nucleotide sequence, in the RNA sequence, there will be alterations, there will be changes. These changes are called mutations. Most of these mutations will not survive. Some of these mutations will provide the virus with some kind of a Darwinian selective advantage, and therefore this virus gains the advantage to spread better or sustain itself well. So what has been done is that from different parts of the world, we have gathered isolates of the virus and we have sequenced them. And these sequences have been placed in an international public domain database. All of you can actually access the data. You can then look at these sequences and do what's called a phylogenetic analysis. So what is phylogenetic analysis? In phylogenetic analysis, we use these DNA sequences and try to group these in this case RNA, try to group these nucleotide sequences into certain number of clusters. And the major idea being that sequences that are within a cluster are very similar to each other, and sequences that are taken from two different clusters are quite dissimilar to each other. If we look at this entire phylogenetic tree, it is really like a tree. It's called a tree. It's really like a biological tree that we see around us, a large banyan tree, for example. It has a base and it has lots of branches. Twigs on the branches on the same branch are similar. Twigs across the different branches may be dissimilar. So that's how the phylogenetic tree is constructed and is known. If we look at the phylogenetic tree of this particular coronavirus, SARS-CoV-2, there are essentially 11 branches, meaning that there are 11 subtypes of the coronavirus. With the ancestral virus, the root of the virus, root of the tree, being the same virus that we actually found in Wuhan. And these viruses, these subtypes have been given different names, O, A1, A3, A4, B1, B2, B3, A2, A, etc. Interestingly enough, and this is where I will end my story. Interestingly enough, what you would expect is that in different regions, you would expect different kinds of different subtypes of the virus to proliferate, to coexist, proliferate, infect other individuals, etc. But and initially that's what happened when the virus moved from Wuhan to Europe. There were many evolved viruses were also proliferating in Europe. But as we analyzed the data, in about 2 months' time, the entire pool of RNA viruses, of coronaviruses, became the same subtype. Although there are 11 subtypes, only one subtype dominated, and that particular subtype outcompeted all of the remaining subtypes and became the dominant subtype across the world. It became the dominant subtype, and this dominant subtype is called the A2A. And we now know, again, like I said, that I'm not going to get into the detail. We now know that this particular subtype is characterized by an amino acid altering mutation, which is called D614G. This means that of the 30,000 nucleotides that there are groups of 3 make up an amino acid. So roughly about 10,000 amino acids, actually far less than that, because not every triplet codes for an amino acid. But anyways, to simplify matters, there are a large number of amino acids. At the 614th position, in the other subtypes, it is the aspartic acid. B stands for aspartic acid. So the other subtypes have aspartic acid at that particular position, 614. But the A2A type has glycine, and G is glycine amino acid at that particular position. So the A2A virus is defined as the D614G type, D meaning aspartic acid or aspartate, at the 614th position that has changed to glycine. So this A2A now has become the dominant virus across the entire world. And many of us were interested in knowing why is it that this particular coronavirus of the 11 subtypes, this particular subtype has spread so well. And if you look at the biology, again, I will cut a long story short. The biology says that if it has glycine at that particular position, 614th position, that glycine is able to anchor to the human lung cell type 2 pneumocyte much better, to the ACE2 protein much better than if you had aspartic acid. So if a virus is able to anchor itself much better on the lung cell surface, then many more lung cells will actually be anchored by the virus, and therefore the chance that this individual will be able to become the home of the virus and then enable the virus to be transmitted, that becomes much more efficient. So this D614G, the glycine residue, the glycine amino acid, provides that advantage to the subtype A2A, in that it is able to outcompete all the other types. It's able to anchor itself better on the cell surface of the human lung cell, and thereafter be able to proliferate well. So I will stop here. Let me summarize then what I said. I mentioned about a few characteristic features of the virus. I mentioned to you about the epidemiology of the spread of the virus and the ancestry of the virus from bats to pangolin to human. I mentioned to you about the spread of the virus and how the virus is spreading, both in terms of its biology and how you track the spread of a virus. And finally, I ended by saying that, how do you manufacture vaccines? What are the major phases of the vaccine, and where do we stand with respect to the coronavirus vaccine, and what it means in terms of controlling the infection, the spread of the infection, in terms of gaining immunity against the coronavirus. I will stop here. Thank you very much for your patience. I'm happy to take any questions.
Thank you very much, sir, for this excellent session. Sir, we have some questions from the participants. Kindly reply to their queries.
I certainly will. I'll try.
Yes, sir. First question is from Parul Banerjee. She asks, is there a chance that SARS-CoV-2 mutates back to a less virulent type, kinder than it is today, like the other four relatives of it, so that human as well as virus both could be able to coexist? Will it be possible?
So this particular virus is not at all virulent. By virulence, we mean its ability to kill people. This virus is a very kind virus. It's killing less than about 1% of the people that it's infecting. This virus is a super efficient spreader. It spreads very well, it transmits very well, but it is not virulent at all. So it's, I don't think there will be one mutation. If there is a mutation, it will make it more virulent, more likely, than less virulent.
Thank you, sir. There is next question from Saman Ghosh. He asks, is 3CL protease responsible for post-transitional modifications of viral polyproteins essential for its survival in the living cell? Is there any other process found for drug-based therapy?
So as of now, I talked to you about this one particular protease called TMPRSS2, and I brought up this protease because it is involved in the process of cleaving. Once the virus gets inside of the cell, there may be other kinds of proteases that we can use in order to kill the virus, and that can be a useful therapy. I honestly don't know about the details of the kinds of treatment therapy. I know a little bit about the vaccine work that's being done. I don't really know about the treatment work that's being done. If it is possible to target any kind of protein against only the virus, that's good. But proteases are dangerous because proteases will also chew up the human host proteins. So you need to be careful in your attempt to chew up some of the viral proteins and kill the virus as a part of a treatment strategy. You might actually harm the individual as well, harm the host as well. So one needs to be careful. But again, this is not giving a direct answer to your question. As a matter of fact, I don't know if protease treatment regimes are being thought through. I honestly do not know.
Thank you, sir. There is another one from Jessica Horo. She is asking, is this virus fatal for someone who is suffering from bronchial allergy?
What is the question then? Bronchial allergy, but what was the question?
She asks whether this virus is fatal for those who have bronchial allergy.
Better?
Fatal.
Fatal. Oh, fatal.
Yeah,
oh, fatal. Oh, okay.
Yeah.
So again, anybody who has asthma, pre-existing respiratory comorbidities, including the kind of allergies that you're talking about, are definitely at high risk. So I cannot say that anybody with a bronchial allergy will be fatal or the virus will cause fatality in such a person, but certainly such a person is at high risk of dying upon infection by the virus.
Thank you, sir. There is one more. If food and water are not responsible for the spread of coronavirus disease, then what are the modes of transmission? They say no food or water can transmit this virus. Is it true? They are asking actually.
So again, whether it is true or not, they are certainly not the dominant modes of transmission. The dominant mode of transmission is droplet transmission, but there have also been cases of fecal or oral transmission of this particular virus. We are not very 100% sure. If it is not a major time to prove that that can also be an alternative mode of transmission. So right now, because it is so dominant, the droplet mode of transmission is so dominant that people are not even bothered about the other kinds of transmission. So, but it takes a long time to gain it.
Thank you very much, sir. I would like to ask Dr. Manisha Kumari, Assistant Professor, Zoology Department, Nirmala College, Ranchi, to kindly extend vote of thanks to our respectable resource person.
Thank you, ma'am. This is my great honor and privilege to propose vote of thanks to Professor Partha P. Majumdar. Sir, I express my heartful thanks to you for enlightening so much about the evolution and spread of SARS-CoV-2 so nicely and generously. It was indeed a great opportunity to hear to you, sir. Thank you once again, sir.
Thank you. Thank you for your patience and thank you for the honor that you've given me to come in front of you and give this talk. Thank you very much.
Thank you, sir.
Thank you, sir, for this session. And thank you, Sister Principal, Dr. Sister Jyoti, Nirmala College, Ranchi, for being with us. Yes, once again, I will thank our honorable resource person. We have longed to hear him and it was a fruitful session. Thank you, sir, very much. And dear participants, our second session will start at 12:15 p.m. Kindly join 5 minutes before. Thank you very much. Respected patron, Dr. Sister Jyoti, Nirmala College, Ranchi. Respected Dr. Kakali Bhadra, Assistant Professor, Zoology Department, Kalyani University, Kalyani. And our dear participants, welcome in the second session of two-day national webinar on pandemic COVID-19, pathophysiology, prevention and treatment. Till date, we know there is no specific vaccine for treatment of COVID-19. However, there are many ongoing clinical trials evaluating potential treatment. Dr. Kakali, Assistant Professor, Kalyani University, Kalyani, West Bengal, is our esteemed resource person for this session. She will be presenting an overview of treatment against COVID-19, synthetic drugs to national metabolites, and overview. I request Dr. Veena Pani to kindly introduce our esteemed resource person. Over to Dr. Veena Pani.
Thank you, ma'am. On behalf of the organizing committee, I, Dr. Veena Pani, welcome our resource person for this session, Dr. Kakali Bhadra, Assistant Professor, Kalyani University, Kalyani, West Bengal. Dr. Kakali Bhadra has been awarded for the Dr. Gurdeep Singh Best Thesis Award in 2010 by Indian Thermal Analysis Society. Her research areas include nucleic acid, small molecule interaction, biophysical and biologic approaches, polymorphism in nucleic acids, binding and cytotoxicity of plant alkaloids, and in vivo in vitro cytotoxicity, insect-plant association, and electroangiography. Currently, she has 2 research projects funded by DS2W West Bengal and DST RFBR, and completed 2 others funded by CSIR and UGC Government of India. Very recently, she has been awarded World Championship 2018 in Zoology for outstanding scientific contributions by International Agency for Standards and Rating. She has more than 50 publications. Today, she will deliver the lecture on an overview of treatment against COVID-19, synthetic drugs to natural metabolites, a retrospective. We are pleased to have you, ma'am. Thank you.
Thank you, Dr. Veena. Ma'am, may I request you to begin your presentation? Over to Kakali Bhadra, ma'am.
Treatment against COVID-19, synthetic drugs to natural metabolites, a retrospective. We are pleased to have you, ma'am. Thank you.
Hello. Hello.
Yes, ma'am. Yes, ma'am.
Am I audible?
Yes, you are very much audible. You can start your talk.
Okay.
Yes.
The slides are also visible to you, I think.
Yes, they are.
A very good afternoon to all of you. First, let me thank the organizing committee and Principal Madam Nirmala College, Ranchi, for giving me this opportunity and for including me in this webinar. Before starting my talk, I would like to acknowledge that I am neither a virologist, not from, I will share with you, is totally based on some recent publications and some featured current informations. So first, let me share a very common information with you all. Nowadays, every one of us gets this message either from any social network or from any newspapers, television channels. Yesterday, even Dr. Moria has also shared a lot of information regarding this against the pandemic from this COVID-19. My question to you is that what the research says about this COVID-19 treatment at this point of time. Today's data, that is on 11th August, I would like to share some values based on some worldometer information that total cases reported in the world is over 20 millions. India has crossed more than 2 millions. West Bengal even has crossed more than 90,000. Even Jharkhand is showing also a number of around more than 18,000. Though Professor Majumdar has given us a lot of hope, but still the picture is not very friendly. It is definitely a gloomy to all of us. I would not like to share any structural detail about this virus anymore after our brief and very lucid information from Dr. Majumdar as well as Dr. Kaviraj that has delivered yesterday. So in a brief and in a nutshell, so this virus, that is coronaviruses, are large family of viruses that causes illness ranging from the common cold to a more severe diseases. This is novel coronavirus. since it has not been reported or identified earlier. As far as its structure goes, detailed structure again I will avoid. So structural protein, it includes few structural proteins are reported, very common structural proteins like spike glycoprotein, membrane protein, nucleoprotein, genomic RNA, that is the single-stranded positively strained genomic RNA, envelope membrane protein, glutamine esterase, and many more. As far as its life cycle goes, already this part has been also covered by Professor Majumdar. But I would like to just give a brief idea that this is happening in this way, that this coronavirus has got a spike protein as S protein. So it binds with the receptor ACE2, which is present on the, am I audible to you all?
Yes,
Yes,
ma'am.
ma'am, you are audible. Ma'am, please start the sharing.
Okay.
आप run कर दो sir मेरे को.
Okay. Should I run from my side?
I think it is okay.
Okay, ma'am. Yes, ma'am, it is started.
Okay. So what I was going, I think here I was, I just stopped here. So before the initiation of the spike, that is the S protein with the ACE2 receptor, actually it is, as Sir was telling even that it is the temprase S serine protease that basically dissociates this S1 and S2 subunit of the spike protein before binding it to ACE2, the human cell receptor. And what happened? It is the initiation of these, after the initiation of this binding, or after binding, the virus accesses to the host cell and is released to the cytosol of the host cell. The viral RNA is translated by the viral polymerase. Following the replication and the subgenomic RNA synthesis, the viral structural protein like the spike protein, envelope protein, and membrane protein are translated and inserted into the endoplasmic reticulum for its post-translational modification, for its further post-translational modification. After that, these proteins move to the endoplasmic reticulum Golgi intermediate compartment, where it coalesces with the nucleocapsid protein. After that, it enters in the Golgi vesicles, and from there it is released outside the host cell. So this is a very brief idea that I'm sharing with you all regarding its life cycle. Now my question is again to you all. Though Sir has given us a lot of hope and called it as a crazy virus, but is there really any cure yet for this pandemic infection by COVID-19? So doctors and researchers are still scrambling to find various techniques to fight this infection, whether it is repurposing drugs, that is the drugs which are already there, or any alternative antivirus therapies, or any natural metabolites that can probably save the infection and save the people from its infective property. But still now it requires its validation and approval. So as we can see that till now, more than about 1,500 pharmaceutical companies got their FDA registered facility, in which only USA comprises about 28%, followed by European unions. India is also not far behind, followed by China, Canada, and rest of the world. But question is again, could these existing drugs really can work on specific symptoms and response against this infection? novel coronavirus has put this drug chemical industry on high alert. It is basically reshaping the pharmaceutical supply chain. And accordingly, I have enlisted a few pharmaceutical companies which are available in India based on their market capitalization. And already among these pharmaceutical companies, few have been credited with some drugs to fight this infection, though they have not received a final approval. Like from Dr. Reddy's laboratory, it has been come with Favipiravir. Cipla Limited is also credited with tocilizumab and remdesivir. Glenmark is with Fevifew, though there is a controversy with this Fevifew. I don't want to go to that. Anyway, people are coming with some drug which we are mentioning as repurposed drugs. Accordingly, I have divided these repurposed drugs under 4 categories. Widely used drugs, that means the treatments that have been used widely to treat the patients. Some drugs under promising evidence, that means though early investigation suggests effectiveness, but still need more thorough research. Some under tentative or mixed evidence, that means the treatments showing promising results in the limited condition, but still well-designed clinical trial is pending. And non-promising drugs, that means treatment showing mixed evidence suggesting that they are not effective at all against this pandemic, that is COVID-19. So our first such repurposed drug or synthetic drug molecule, remdesivir, it was first described in the literature in 2016 against the treatment of Ebola. In 2017, its activity was reported against various coronavirus family, and later it was found to be potential against Sars Cov-2 also. But it was granted an FDA emergency use authorization, but not yet has received any approval from the FDA. Regarding its mode and mechanism, remdesivir is a prodrug of an adenosine triphosphate analog with potential antiviral activity, as I have mentioned. Upon administration, this remdesivir is converted into its active form, metabolically active form, which is basically the ATP analog, act as an ATP analog. And it competes with the ATP, that is a normal nucleotide of the virus, for incorporation into the RNA, and inhibits the action of viral RNA-dependent RNA polymerase. And this results in the termination of RNA transcription and decreases the viral production. But here is a doubt that this virus has been reported to have a very good proofreading mechanism using at the expense of various endonucleases. So in that case, how far this error will be avoided by the virus, it's a big question mark. Next one, that is Favipiravir. Like the experimental antiviral drugs, it is a Pyrazine carboxymite derivative. It was commonly named as Fevifew, and it was first invented by Japan and was treated against influenza. It is still under trial for phase 3 trial, and it selectively inhibits RNA polymerase. EIDD-202801 is another experimental antiviral drug which is orally active and was developed for the treatment of influenza. It is a prodrug of the synthetic nucleoside derivative N4-hydroxycytidine and exerts its antiviral action through the introduction of copying error during viral RNA replication by mimicking the ribonucleoside. So almost it is similar in its mode and mechanism of action like remdesivir, but it still requires its approval. Next, the chloroquine or hydroxychloroquine is a much talked and very known drug molecule everyone is acquainted with. So basically, this is the structure of the chloroquine that this R is replaced by OH and it becomes the hydroxychloroquine. As we can know that already so many pharmaceutical companies, including India and abroad, are engaged in synthesizing this molecule. But question arises that how far it is effective at today's scenario, or what is the current status of this hydroxychloroquine? Basically, hydroxychloroquine was functionally active, but it is used against rheumatoid arthritis. And basically, this drug was used in combination with another synthetic molecule called as prednisone, which is basically the glucocorticoids. And this basically reduces the pain or swelling and inflammation that happened in the rheumatoid arthritis. So its current status, based on its ongoing analysis of the EUA and emerging scientific data, the FDA determined that hydroxychloroquine is unlikely to be effective in treating COVID-19. The reason? Because of its severe cardiac effect, hepatic toxicity, its liver toxicity, and many other adverse effects. But if we have to know its mode and mechanism of action, it is like this. Hydroxychloroquine enters and accumulates in the lysosomes along the pH gradient, where it inhibits the degradation of the protein derived either externally or internally in the autolysosomes by increasing the pH. So this is its most important mechanism, that is by increasing the pH. So inhibition of lysosomal activity can prevent the MSC class 2 mediated autoantigen presentation. HCQ can also accumulate in endosome and can bind to the minor groove of the double-stranded RNA. Why it is with a minor groove? As we can see that the structure is not typical of planar molecule, hence it cannot intercalate directly into the DNA base pair. So it acts as a group binder or minor group binder. And its second mechanism is that this drug can even inhibit the toll-like receptor signaling by altering the pH of the endosomes or by even preventing the TLR7 or TLR9 from binding their ligands. So in nutshell, what I can tell that in both the cases, it is hampering or it is changing the lysosomal pH and it is preventing the viral enzymes to act in this pH media. Next, another synthetic molecule which is always taken in combination, that is a lopinavir and ritonavir. The reason of taking this ritonavir is basically taken in a very low dose and it basically enhances the longevity of this lopinavir. This was used against HIV and AID, that is AIDS. These are antiretroviral of the protease inhibitor class. And it was first patented in the year 1995 and approval was received in the year 2000. It has got many side effects like diarrhea, vomiting, headache, muscle pains, and chronic effect includes pancreatitis, liver problem, and high blood sugar. And its status is not at all found to be promising against this COVID-19 infection. Dexamethasone, it is also a very commonly known drug molecule. So it is a synthetic adrenal corticosteroid with potent anti-inflammatory properties. In addition to binding to specific nuclear steroid receptors, dexamethasone also interferes with the NF-kappa beta activation as well as apoptotic pathway. So its approval is not very fruitful and it has not been recommended against this infection. So this is one more synthetic molecule very recently reported. I got this information from one publication published sometimes in the month of July. So what they had showing that based on molecular stimulation, they are showing that this alpha ketamide might act as an inhibitor against 3CL-Pro or 3-chymotrypsin-like protease against this SARS-CoV-2. So what is this 3CL protease? So it is basically play an important role in the processing of polypeptides translational product from the genomic RNA into various structural as well as the non-structural protein. But again, as Professor Majumdar was telling, this is a very true fact that proteases are also harmful for the host cells also. So for this, it required very efficient in vitro study before it received its approval. So the next one is the semi-synthetic compound. This is again, I got the information based solely on the molecular stimulation, ivermectin, that has been reported to bind in the inter-subunit cleft of the S protein. So at this point of time, none of these repurposed drug has received any approval from FDA, and its status is really at stake against this infection. So hence I have decided, or we can think of some alternative or other therapeutic action against this COVID-19 treatment, like convalescent plasma therapy. So this was already explained a bit by Dr. Sharma yesterday. Stem cell therapy. So basically these two therapies are called as rejuvenating medicines. Why? Because they are rejuvenating damaged body tissues. And the other four, like recombinant ACE2 therapy, interleukin-6 inhibition, interferons therapy, or cytokine inhibitors, etc. So let's explain the convalescent plasma therapy. So basically the blood from the people who have recovered from the infection is called as convalescent plasma. So convalescent plasma has not yet been shown to be very effective against COVID-19, though it has been found to be very effective against other viral infection like influenza or Ebola infection. FDA states that it is important to determine its safety and efficacy by a clinical trial before routinely administered to patients with COVID-19. Its exact protocol and use is still under research. So in a nutshell, basically this convalescent plasma contains a mixture of antibodies. Only some of the antibodies can fight this virus. Basically, they are the monoclonal antibodies that is supposed to fight against this infection, against this COVID-19 infection. So stem therapy, which is again another rejuvenating medicine. The difference with this convalescent plasma therapy is that it is a basic generic building blocks of life. And in a sense, they are unspecialized, undifferentiated immature cells. They can divide and differentiate into specific types of cells and tissue based on specific stimuli. They can be formed from cord blood or bone marrow. They have got various functions, and in taking consideration or considering those functions, people are thinking about using this as a therapy against this COVID-19 treatment. Like certain types of stem cells can secrete anti-inflammatory molecules. It can also produce certain growth factors and cytokines that can accelerate the healing process at the site of tissue damage. It is used to treat degenerative and inflammatory conditions by replacing the damaged cells in virtually any tissue or organ. But researchers have tried to use them to treat cytokine storm, though it has been found to be helpful. But now dozens of clinical trials needs to be ready before it gets its approval against this infection. Another therapy is the recombinant ACE2, as I have explained, the ACE2 angiotensin converting enzyme 2, which is the human cell receptor with which the spike protein binds. Basically, it is a zinc metalloprotease and has got zinc binding motif. So the recombinant or artificial ACE2 protein might be able to lure its way from the vulnerable side. Taking this into consideration, people are thinking about its therapy. They have shown many more promising results in vitro experiments, but not in any animal model. So it is still under clinical research. And one more point let me share with you all is that Very recently, it has been reported that this ACE2 has been reported to show around 317 missense single nucleotide mutations. And based on, though it is based on molecular stimulation study, but still that factor we have to take into consideration before using this as a therapy against this infection. IL-6 inhibitor, basically IL-6, that is interleukin 6, is a powerful pleiotropic pro-inflammatory cytokine. This is a very specific property of this particular interleukin. It is a major sources of this interleukin are the macrophages, endothelial cells, synovial fibroblasts. So after targeting its specific receptor, IL-6 starts a cascade of signaling efforts, mainly associated with the JAK and STAT activation pathway. I don't want to go in detail about this activation pathway, stimulating acute phase protein synthesis, as well as the production of neutrophil in the bone marrow. It supports the growth of B cells and is antagonist to regulatory T cells. Sars Cov-2 infection induces a dose-dependent production of IL-6 from the bronchial epithelial cells. So when it goes beyond some level, that is optimum level, under severe condition, it becomes severe for the patient. Hence, IL-6 inhibitors are generally suggested. There are various IL-6 inhibitor already reported, that is, they are all pharmaceutical analogs, but not against this COVID-19 infection. So its approval is still at stake. So NIH guidelines note insufficient data to accept it as an accepted therapy against this infection. So this is the, in brief, the JAK and STAT mechanism. So another therapy, that is the interferons. They are basically the molecules that our cells produce to make the immune system attack the virus. So what it happens, virally infected cells, from here, INF gene is activated. They secrete INF protein, that is interleukin protein, which in turn bind with the receptors of the non-infected cells, and which make these non-infected cells to secrete or activate various protein kinase and endonuclease, which will not only kill the virus, but it will also hamper the host genome cells, that is host genome also. So injecting this synthetic interferon is now a standard treatment for immune disorders. Experiment in mice and cells suggest that interferon injection could be used both as preventive and treatment against COVID-19. Already some synthetic interferons are repeated, like pregintron, Pegasus, though this has not been found to be effective against COVID-19 till this point of time. Another one, that is the cytokine inhibitors. The body produces molecules called cytokines to fight the diseases under normal conditions. In excess, they can trigger a condition called cytokine storm, which causes the immune system to overreact to infection. Various drugs are already there which can fight this cytokine storm, like tocilizumab, sarilumab, etc. And various signs and symptoms of these cytokine storm is that high fever, respiratory distresses, inflammation, severe fatigue, nausea, etc. So this is a schematic presentation to show this cytokine storm. So when this viral load in the infection site increases, they will stimulate this dendritic cell macrophages to secrete some pro-immune cytokine factors that diffuses into the blood cells, making them to secrete even more cytokine factors like lymphocytes, monocytes. So they will ultimately come to the infection sites, and at this severe state, it will lead to the production of bronchoalveolar lavage fluid that basically causes the respiratory distress in the patient. So in a nutshell, I can tell you that SARS-CoV-2 is basically a severe condition causing the hyperinflammation. That in turn leads to cytokine storm formation or condition. And as we can see from these conditions, that inflammation basically is a double-edged sword process. Both the inflammatory immune system and anti-inflammatory immune system exist in a friendly way, that is under homeostasis. And the moment this homeostasis is breakdown or broken, that lead to the formation of cytokine storm, and that happens in the SARS-CoV-2 or SARS-CoV-2 infection. Even nowadays, a hemodialysis or hemodialyzer has been invented called as CytoSor, and that basically filters the cytokine, and it has been found to be very effective. So at this stage, we can still tell that neither any specific drug nor any therapeutic approaches has been found to be effective against this infection. So at this stage, can't we think of some natural metabolites which are more easily achievable and more easy to work on? So based on this, I have selected some natural compounds like flavonoids, flavonoids, various terpenoids, quinone derivatives, as well as some natural alkaloids. So what they basically do, they basically inhibit either AC2 transmembrane protein or TMPRSS2 transmembrane protein of the host, or by inhibiting the 3CL proteins. So this action or mode of action and these natural metabolites I have collected specifically against this SARS-CoV-2 infection or SARS-CoV-2 virus. So here are some natural alkaloids extracted from veratrum nigram, which has been reported. So sorry for this interruption, I'm extremely sorry. So as I was telling that they are the natural metabolites. So they have been found to be found to inhibit various transmembrane protein of the host like AC2, TMPRSS2 protease, and also with the 3CL protease action, inhibitory action. So the next one, next slide, please. Next slide, please. Next one. So this part also I have already covered. So next one. Yeah. So these are some of the natural metabolites I was discussing. So based on their molecular docking study, so it has been found to be a very effective inhibitor of AC2 and TMPRSS2 inhibition. And one more thing, let me share with you all. Seeing the structure, I can tell that these kind of natural metabolites, or whatever it is, the metabolites, have been characterized to have some hydroxyl group. They should have some aromatic ring as well as some carbohydrate moiety to be accepted as a very good inhibitor against these two inhibitors, that is the transmembrane protein. Next slide, please. So they are again two terpenoids with that is bonduselpine B and sisselmine B, which has been found to be a very effective inhibitor against 3CL protease. In addition to these two terpenoids, some mono and sesquiterpenes like geraniol, linalool, beta-farnesene has been also found to be very effective against the inhibition of 3CL protease activity of the virus. Next slide, please. Next slide, please. Here are some more natural metabolites reported, and they are all mostly from our flavonoids. and they have been suggested to have a very good inhibition against 3CL protease action of Sars Cov-2, like curcumin, dimethoxycurcumin. They are the most important one, and these are an important constituent of turmeric, as we know it. Next slide, please. So this is another glycoside flavonoids. So this has also been found to be very effective against SARS-CoV-2 3CL protease action. Though some in vitro studies has been done in Vero E6 cell lines, but still it needs further approval and very detailed in vitro study. Next one. Next one, please. This is again another alkaloid, that is cyfarenthine, which is a bis-benzyl isoquinoline alkaloid. It has been extracted from tuber Stephania japonica, and its mode of action has been reported to be against spike protein inhibition. One more compound that is a flavonoid, theoflavine, which is a very good constituent of black tea, and its mode of action has been reported to be inhibitory action against RNA-dependent RNA polymerase activity. Next one. Next one, please. So another natural metabolite called as coumarin, whose main source is from beans, cinnamon, green tea, carrots, and its mode of action is by inhibiting 3CL protease action, though its dose is not very known at this point of time. In addition to these natural metabolites, there are many reported traditional Chinese medicinal formula has been reported by many workers or researchers, which I don't go into detail since their constituent is not still fully known. Next slide, please. Next slide. So till this time, I have the metabolites that I have shared, they were mostly specific for SARS-CoV-2 infection or SARS-CoV-2 inhibitory mechanism. But in addition to this, many more natural metabolites are also there, like imodine, celestriol, and many more. And their mode of action has also been found to be almost similar to SARS-CoV-2. Next slide, please. Next slide. So these are some of the antimetabolites which has been reported to act against anti-SARS-CoV-2 natural or anti-SARS-CoV-2 infection, like isotheoflavine, G-gallate, tannic acid, ethanolic extracts of phenylalanine, lycorine. So these are all basically act against various coronavirus family, but not specifically against SARS-CoV-2. Next slide. I think there is one more. I think there is one more slide before that. Yes. So at this point of time, or my main objective of sharing all these natural metabolites along with their structure is that though they have been not found to be very effective against this SARS-CoV-2 infection, but based on their structure, probably these can be used in a framework to design novel corona compounds for improved therapeutic applications in future against this COVID-19 treatment. So next one, please. Next slide. So again, back to square one. So at this point of time, it is better to say that prevention is better than cure. So boost your immune system, go for food rich in high vitamin C, zinc, and vitamin D, maintain social distancing, and go for workouts. So probably this can boost our immune system so that for time being, we can fight against this virus. Next slide, please. Next slide, please. So these are some of the references with the help of which I have collected the informations. Next one. Next one. So this is the continuation of the references. And the last slide, please. So at this point of time, I must say that every crisis leads to some opportunity. Let's encourage and reward our researchers and medical professionals for their future outcome. They are truly the connecting link, sacrificing their life to fight this infection. Till such time, stay safe and healthy. Thank you all. Sorry for this interruption.
Yes, ma'am. There are some questions for you, but it was a wonderful session and valuable knowledges that you have shared. We are really thankful to you for all these. Ma'am, there are, of course, certain questions asked by our participants. Kindly reply to their queries. First question is...
I will try.
Yes. Okay. It is by Dr. Jaya Rajalakshmi. She asks, what are the side effects of hydroxychloroquine? Does it affect immune system?
Madam, I have already explained this. Basically, hydroxychloroquine is a drug which has been recommended against rheumatoid arthritis. So, along with another drug that is prednisone. Basically, prednisone is the glucocorticoid that fight against the pain and the inflammation that occurs because of this arthritis process. Now, hydrochloroquine as such, how far it should be recommended for COVID-19 or not, this is a big question mark. And regarding its severe effect, yes, I have that part also I have discussed. It has got really very severe, extremely severe effect against hepatic toxicity, against lung, against kidney toxicity, against various cardiac side effects. So at this point of time, I think better without consulting any medical physician, you should not go for hydroxychloroquine against this treatment, against this infection.
Thank you, ma'am. Yes, ma'am, you are clear. Ma'am, there is another question from Dr. Soman Ghosh. He is asking why azithromycin is promising for COVID-19 infection, though it is an antibacterial drug. Is favipiravir a promising antiviral drug or agent against COVID-19?
Madam, I think there are two questions. One is regarding azithromycin, another is favipiravir.
Yes.
Regarding azithromycin, let me tell you, frankly speaking, azithromycin is a very commonly drug people take for any sort of throat infection. Even I have also taken 2 to 3 months before. So this is a very common drug people take, not only for this COVID-19 I'm talking about, any sort of viral infection or any, rather more specifically, any sort of throat infection people take. So now, if physician suggests maybe this azithromycin can be found to be effective under controlled Sars Cov-2 infection, but then physicians should be consulted. But this azithromycin is found to be effective in other viral infection also. Second part is the favipiravir. It still needs its approval because favipiravir is basically used against influenza. So it has not yet approved its FDA approval. So regarding its treatment against this infection, I really don't have much opinion. Any more question?
Yes, ma'am. Thank you for this question's answer. Now another one is from Paramita Bhattacharjee. She asks, according to your observations, which type of immunogenic drug can be the most potent drug for controlling corona infection in our country?
Okay. So, Paramita, at this point of time, really any specific immunogenic drug against is really a question mark. So really out the whatever repurposed drug I have shared, neither of them has got any specific action against this SARS-CoV-2 infection because it has got many other side effects also. So taking everything into consideration, at this point of time, none of these drugs has been found to be very much effective against this SARS-CoV-2. Is it clear?
Yes, ma'am. Everything is very clear. Thank you very much. Now, may I request Dr. Veena Pani to propose vote of thanks to our honorable resource person, Dr. Veena Pani.
Okay. Thank you, ma'am. On behalf of the organizing committee, I extend sincere thanks to our resource person of this session, Dr. Kakali Bhadra from Kalyani University, West Bengal, for her informative talk. We came to know about various synthetic drugs and its mechanism in treatment of COVID-19. You also explained very well about natural metabolites' role against the COVID-19. Ma'am, we are all benefited from your valuable lecture. Thank you so much for sharing your knowledge with us. Thank you, ma'am.
Thank you, Dr. Veena Pani. Thank you, ma'am. Dr. Kakali
Thank
Bhadra.
you again.
Thank you very much. And I also thank our patron, Dr. Sister Jyoti, Principal Nirmala College, and all our dear participants for being with us in this session. We will be back at 3 p.m. Kindly enter the meeting half an hour before. Have a nice day.
Thank you.
Thank you, ma'am. Respected patron, Dr. Sister Jyoti, Principal Nirmala College, respected resource person, Dr. Kasil Kumar, and dear participants, a warm good afternoon to you all. This is the third and concluding session of two-day national webinar on pandemic COVID, pathophysiology, prevention and treatment. COVID-19 has potential to cause devastating health effects. In order to prepare, respond, and recover from it, we need to enhance our immunity. We have with us Honorable Resource Person, Dr. Kasil Kumar, SMS and Head, Department of Forest Products and Utilization, Birsa Agricultural University, Ranchi. He will be presenting on cultivation and processing of some immunomodulator medicinal plants so that we will be able to know the vegetation around us, which is helpful to keep us safe during this COVID period. I request Dr. Indu Kumari, Assistant Professor, Botany Department, to introduce our Honorable Resource Person. Over to Dr. Indu.
Thank you, Dr. Emma. A very good afternoon and a warm welcome to our Honorable Principal, Dr. Sister Jyoti, and a Vice Principal, Dr. Sister Shobha, our respected guest, Dr. Kasil Kumar, and my dear colleagues and participants. The topic of this session is Pandemic COVID-19, Cultivation and Processing of Immunomodulator Medicinal Plants. Dr. Kasil Kumar is Head of Department of Forest Products and Utilization, Faculty of Forestry, Birsa Agriculture University, Ranchi. He completed his PhD in Medicinal Plants from BHU, Varanasi. He has published more than 70 research papers in national and international journals, published many books, delivered more than 60 lectures, and attended national and international seminar, symposia, and workshops. He has been principal investigator of several research and extension projects. He has widely explored medicinal plants through field survey on Himalayan region, Andaman and Nicobar Island, Chotanagpur Plateau, and Indian Desert. He has worked as a senior scientist in Patanjali Yogpith Haridwar. He has been awarded FES, FBS, FNRS, etc. of various academic societies. Now, I would like to request our guest, Dr. Kasil Kumar, to introduce this session. We welcome you, sir.
Thank you. Namaste. And very good afternoon to our principal, Dr. Jyoti, Sister Jyoti, Dr. Irma Madam, Vice Principal, and Dr. Hindu, and Dr. Anubhuti, and all the delegates. We are heartily thankful to all of you that you are very much have attention to understand the problem and constraint of COVID-19. Am I audible to all of you? I am audible to all of you?
Yes, sir, you are.
Yes, sir.
Yeah. Nice, thank you. And this is I think it is the last session. So please enjoy the session as I am talking exclusively for the medicinal plants used as a immunomodulator medicinal plants, exclusively for the COVID-19. And the topics which I have chosen, the cultivation and processing of immunomodulator medicinal plant, because we cannot, as the vaccine has not been developed or released or launched, therefore there is the only sources to protect ourselves. It is the only through the immunomodulator medicinal plant, because it has been already mentioned in Ayurveda, Unani, Siddha, homeopathy, and it is by the Ayush department. And it is the one of the thousands and thousands years of our tradition, culture, innovation, and practices through the home remedies, through our folklore tradition, through the Ayurvedic tradition, through the all kinds of reverse pharmacology validated condition. So now, as there are a large number of requirement of the medicinal plant to prepare the herbal product, so now the time is for the cultivation and processing. As everybody knows that according to the WHO, World Health Organization, medicinal plants is widely used for the primary health care, and it is the one of the most safe kind of treatment. As I am working on the Department of Forest Product and Utilization through the Birsa Agricultural University, and we are already doing particularly on the research and extension work exclusively for the cultivation of medicinal plants, promotion of medicinal plant among the farmers in the Jharkhand and adjoining states. And there are a large number of farmers out of the Jharkhand. They are contacting with us, and they are really interested to understand about the which kind of germplasm, what is the cultivation practices and packages, what will be the post-harvest technology, what will be the value addition, what is the marketing. But now in perspective of COVID-19, there are certain medicinal plants which are really found very much effective, as well as people are still utilizing, and they are already mentioned in the text of the Ayurveda. See, this is the practices of non-timber forest product, medicinal plant, raw material. It is a view of a local market. It is usually sold in the market. There is a large number of raw materials, but the wisdom or the science or the hidden there principle of the activity. Now there is a lot of tremendous demand in the global market on the quality planting material. So this is one of the important aspects to understand about the scientific cultivation practices, their agro-technology. I will not talk about such kind of plant which are new. It is already been mentioned in several kind of literature. It is already validated through the ethnopharmacology, reverse pharmacology, mentioned in Ayurveda, and a large number of medicinal plants are utilized by the pharmaceutical industries for the preparation. So if this session is very important to all of the viewers and listeners, because I will introduce with some medicinal plants which are utilized thousands and thousands years of the innovations of in the local health tradition, and it is a very good and safe immunomodulator. People will ask, what is the importance of the medicinal plants? Suppose somebody says medicinal plant is the medicine, but the constituent which are found among the medicinal plants parts, that is the medicine. So I will go discuss one by one. So the plant constituent, the major groups are alkaloid, steroid, phenolics, tannin, glycoside, glucoside. And around the world, there are a large number of examples of some constituents like aconitin, quinine, azadirachtin, aprin, vincristine, vinblastine. This is the anti-cancer, asiaticocyte. It is the very good Nirmala, curcumin from the haldi, digitotoxin, very good cardiotoxin. This is quinine, reserpine, somniferin, vidinin, it extracted from the ashwagandha, andrographoloids, eugenol, menthol, piperine, caffeine. So it is some example, some constituent of the medicinal plants which are very widely used around the world. And the entire now looking to the India, entire world is now looking to India to know about the which kind of medicinal plants are important, and India which plants are promoting. So this is very much importance of the medicinal plant, and it is not just only a folklore or home remedy. It has been scientifically proven through the ethnopharmacology or reverse pharmacology, and there are some of the large number of secondary metabolite components are already in the market. So there are no problem, and their toxicity, safety, efficacy has been already evaluated. But the COVID problem, unique problem, and it has been never been earlier happened, and the entire world is now facing such kind of problem because vaccines are not at all. And that this particular medicine is good, very much effective. So there is a lot of opportunity to manage oneself through the immunomodulator medicinal plant. Somebody asked, what will be the basis of reported usage of medicinal plant? So this is very much in the context that this is described in the classical text. Suppose in the Ayurveda, there is an ancient Ayurvedic wisdom. Now it is already validated through the Ayurvedic text, Dravyaguna. There is several Nigantu, Charak, Susrut, Bhagavat, Ashtang Hridaya, Bhaprakash Nigantu. And there are large number of scientifically validated medicinal plant texts. Those are described in the classical text. Likewise in the Unani system, there likewise in the Tibetan system, likewise in the Siddha system. Second, the folklore or ethnobotany. As we know that the medicinal plant used by the tribal people, indigenous people, native people. Suppose in the case of the Jharkhand, there are a large number of tribal people like Santhal, Paharia, Aura, Munda, Kul, Kharwar, Ho, Asur, Bega. That's all are utilizing the medicinal plants in their own healthcare tradition. And it has already been documented and published by us. And there are a large number of research scholars from the several institutes like Botanical Survey of India, National Botanical Research Institute, from the several universities. There are a large number of theses that have been published. So some medicinal plant has not been validated or evaluated. It is another thing, but it is mentioned in the folklore or ethnobotany and different system of medicine. Thereafter, it is the proven through the pharmacology or clinical trial. There are a large number of medicinal plants which are used by the tribal people. As well, it has been also mentioned in the classical text of Ayurveda. Suppose there is a bang sag in the Jharkhand, that is the Centella asiatica. It is also used by the Santhal people as a rote ada, bang sag. But it is the Centella asiatica, it is the Manduparni. And it has been utilized for as a nervine tonic, brain disorders, for the memory. So it has been considered. And the pharmacology, toxicology, there are, suppose in a specific dose, some plants are found to be very much good, effective and safety. But in the large quantity, of taking of large quantity beyond the recommended dose, it becomes toxic. So through the toxicology, their lethal dosage concentration and validation of the claims. There are certain claims appear, but it should to be validated. So in this context, some plants I am taking on the basis of the reported usage of the medicinal plant. So there is now need of agency or guideline of quality medicinal plant. There are certain agency in the internationally, that is the World Health Organization, WHO, who has published several kind of quality control. There are good agriculture practices, good manufacturing practices, what will be the safety measure. Second thing is the pharmacopoeia, like Ayurvedic pharmacopoeia, Unani pharmacopoeia, and around the world there is American pharmacopoeia, Chinese pharmacopoeia. On every country has the different situation of cultivation. Every country has several kinds of parameters according to the agroclimatic conditions, the zones, and the particular of the plant species. Suppose a population of a species growing in different environmental conditions having different therapeutic efficacy. Because it is the separate science of the biosystematic and medicine. A medicinal plant which are occurring from the different biogeographic region may have the different kind of effect. So the pharmacopoeia has a particular standard that in a particular parameter, some raw materials, what will be the effective. Now the National Medicinal Plant Board, it is also one of the important leading organization of Government of India for particularly control or promote the medicinal plant materials. Now, in perspectives on COVID-19, suppose there are a large number of problems already has been taken very good, taken initiatives and for the treatment through the traditional healthcare medicinal plant, through the Ayurveda, Unani, and through the folklore. But now the emerging situation is COVID-19, how we can manage. So there are certain medicinal plants which are very good, found effective as a immunomodulator to protect ourselves through the certain principles. So it will be discussed. So immunomodulator medicinal plants. Immunomodulator medicinal plant, what is, it is all through the immune system. It is immunosystem. It at first on cellular immunity. Second thing, antibodies mediated defense system. This may be defined as the body's ability to identify and resist large number of infectious and potentially harmful microorganisms, enabling the body to prevent or resist disease and inhibit organ and tissue damage. Although immunomodulator or immune system, the definition is very vast and complex. It differs on particular person to particular individual. But the immune system is not confined to any part, one part of the body. It is overall mechanism. These are biological or synthetic substances that can stimulate, suppress, or modulate any aspect of the immune system as adaptive. some of the medicinal plants valued in Ayurveda Rasayan for their therapeutic potential have been scientifically investigated with promising results. A number of plant-based principles have been isolated with potential modulatory activity that can explain and justify their use in traditional medicine. So now it is the best time, it is the best option, it is the best period to adopt immunomodulator medicinal plant, which are the Ayurvedic science, maybe as effective in other system of medicine also, but as we are residing in the India. And it has been already mentioned that such kind of medicinal plants are Ayurvedic Rasayana. And it is very good rejuvenating of our cells. It may be used for the cell repair. These all are antioxidant. So such kind of usage of medicinal plant is very much found to be effective. And there is need for large scale cultivation, because suppose now people are regular interested to cultivate the medicinal plant for the good quality planting material. Now, the concept is Jaivik cultivation based medicinal and aromatic plant, because organic based cultivation, not inorganic, because Gaya ka gobar and Kechua khad. This is the important aspects of the Jaivik cultivation. There should be not any contamination with the organic manure. It should be purely, fully purely the cow dung, FYM, and the earthworm. Because Jaivik cultivation, where there is no earthworm in the field, it means there is no Jaivik kheti. It is organic cultivation also. It is already known as a organic cultivation. But in Hindi, as organic cultivation, the simple Hindi is the carbonic kheti. But in Indian tradition, it has been adopted as Jaivik kheti. Means it is some vitality. It has to vital produce. So there are certain differences between organic cultivation, carbonic krishi, but Jaivik kheti, it is one kind of vital cultivation. So there is a great aspects of Indian tradition for the Jaivik cultivation. So now in the entire Indian pharmacies has started to take or procure the organic cultivated material, organic produce. So if some farmers or entrepreneur or any kind of medicinal plant grower who are interested for the cultivation of medicinal plant, they have to understand the principle, logic and theory of the organic cultivation first. Because inorganic contamination, the quality and the safety and efficacy all will be affected. Now, scientific ethics of the cultivation we have to understand. To control quality, to get desirable yield. Suppose if somebody is saying that I will collect from the forest, I will collect from my garden, I will collect from the animal, it is my choice, I will collect and wash and just use. No, because there are certain contamination of the heavy metal. Suppose there is a mining area, there is some coal field, there is some arsenic zone, there is some chromium zone. So we cannot collect any of the medicinal plant randomly from the forest. It is a very good strict provision, because there will be adulterants, there will be no safe, because it may be from the polluted area, it may be from the mining area, and suppose somebody, but if you are going to cultivate by your own practices in some field, in some controlled parameter conditions, there will be manage of the secondary metabolites, their chemical constituent. It will be maintained through all the parameter. It will be not contaminated through the either environmentally or through the any kind of deposits of the heavy metals. Suppose there are several medicinal plant or herbal product are being rejected. If it is found that there is some contamination through the arsenic or some lead or some chromium or any kind of heavy metals, it affected. Now, suppose there is no earthworm in the field, it means it is not organically cultivated, because earthworm is only survive if there is not any kind of inorganic contamination. So it is the only of the best parameter. So to control the quality, to get desirable yield, it is strictly prohibited adulteration. It is a sustainable utilization. Sustainable utilization, what is the? It is a part of the conservation. The definition of conservation is always that conservation is the preservation for sustainable utilization for future generation. So sustainable utilization is necessary because there is some 4 or 5 samples if available in the forest. People are going and collected. Therefore, the germplasm will be vanished from the forest. So the sustainable utilization is important aspect. And to protect the wild genetic resources, there is need to protect the wild genetic resources, because it is evolving through the environmentally, genetically, and in the generation to generations, and it will take time. But there are certain varieties, if suppose released by the scientific institutions, there are many varieties have been released by the, like many CSIR organization, NBPGR, Agricultural University, ICAR. So there is need of the cultivation in a control parameter. So it will be not possible to collect the plant from the natural resources, but it is the ethics of the cultivation. And I will request to the participants, they have to note some points, and ask at the end of my lecture there, so that I can help him to solve their problem. Now, herbal industry reaching in top, because there is now the whole world, herbal and digital. The two platforms are very much in the high job, because herbal has the very much safety and it is useful for the mankind. And now the most of the cosmetic products, now the healthcare products, primary healthcare products, due to the therapeutic, curative properties of the medicinal plants. And the medicinal plant, it is a symbolic, herbal is a symbolic to the medicinal plant. It represents the word, it is just medicinal plant or herbal. It is both are the, maybe used by the parallel by the general public, because as per the WHO and other agency, it has adopted that the herbal is a term, and the definition of medicinal plant is at least one part of the plant, if contains medicinal properties, may refer as a medicinal plant. Suppose haldi is a spices, but it is a good medicinal plant because it has some medicinal plant efficacy for the wound healing, for the allergy, for the cough and cold. So although it is a spices, likewise gol mirch, the piper nigrum, but it is also spices as well as medicinal plant. Now herbal industry reaching in top, regularly their turnover are increasing, and there are a large number of herbal industry are regularly promoting the medicinal plant. See, people are generally believe that if some products are from the medicinal plant or herbs, it will be just like any kind of crushing or their decoction or their extract. No, now the herbal industry has a very sophisticated instrument. It is a view of the boiling chamber. It is for the extraction method. There is a large number of good quality of use of internationally utilized like pulverizer, grinder, extractor, juicer, pulper, boiler, and starting from the purification or cleaning of the raw material to end of the finished product, because in the two stages there are need to be quality control. First, in the raw material stage, suppose some raw material came for sold in the public domain for the industry. Industry are first taking investigation about their utility as per quality parameter. So the quality parameter has been evaluated at the raw material stage. Second, it may possible that some contamination may be hidden in the product. So it is at the end of the finished product. Suppose raw material has been used for the making herbal products. Now it may be possible that in the it has been not checked very highly, very authentically the raw material. Then in the finished product, if some contamination is present, then through the atomic absorption spectrophotometer, other kind of sophisticated instrument, it will be checked for the heavy metals. Like HPLC, HPTLC, mass spectroscopy, NMR, GCMS, all kinds of instruments are utilized for the evaluation of the good quality of raw material. Now, the WHO guideline is the good agriculture practices. Good agriculture practices, and second is it will be bias and versa good manufacturing practices, because effect of herbal products is always depends on facts of quality of herbs in the preparation. So in the preparation, if some herbs are not quality, having some deterioration, having some contamination, having not up to the parameter, so it will certainly affect the herbal products. As raw material will be supplied, products will be affected. So it should to be good agriculture practices, because they are in the good agriculture practices, which is a scientific agriculture practice, not in the haphazard manner. So just to cultivate without and pushing any kind of, so in a control their nitrogen, phosphorus, potash, or now the use is the neem, karanj, and other kinds of their cake for the organic cultivation. So manure for their collection practices in the what will be the season, how you will store the medicinal plant in a controlled condition. And so it is important to understand the effect of herbal product is always depends on facts of quality of herbs in the preparation. Next. So it is now the processing of any medicinal plant, it is fully scientific methods. Suppose it is the HPTLC. You take 20 ml, 10 ml, 5 ml of the sample, and anybody can analyze scientifically what is the chemical constituent, what is the component, because now these days you can say anything about the importance of medicinal plant, but nobody can believe if you have not scientific evidences, if you have not any kind of publication, if you have not the data, clinical data, pharmacological data, toxicological data. So somebody if has mind that somebody is telling about just to promote the medicinal plant, it is wrong, because nobody is going to believe if there is some scientific methods are not has been published in the public domain. So the medicinal plants, the work on medicinal plants is fully scientific methods, and there are a large number of publications and which has been already published and already adopted in the pharmacopoeia standard. There is also come for the proprietary medicine, then it come in the public domain. The scientific method is the secondary metabolites, as I have earlier told, that the plant active principle or the medicinal plant constituent which are important has been isolated. There is a protocol of manufacturing, and this view is of an Ayurvedic industry. You may not believe that now the pharmacies of medicinal plant were all kind of sophisticated instrument for the analysis of the medicinal plant, and it is necessary, it is mandatory that they have their R&D. So any kind of medicinal plant, if somebody are interested to cultivate, they have to go for the good scientific methods for the cultivation, because secondary metabolite will always affected and plants will be not efficacious. So these are one of the, and now entire world is now searching the solution of medicinal plants, the sources of medicinal plant for the treatment of diseases and disorders. Now I will say about the one by one, which is the best immunomodulator, and which are the in the entire India and around the world there are discussion are ongoing that which plant is the best or how much plant is the important as a best immunomodulator. So I will say that there are several government agencies as well as the private pharmaceutical industry as well, and already described in the Ayurveda. This is the Giloy, Tinospora caudifolia. It is the Guduchi. You know, Guduchi in the Ayurveda is said that is the Amrita. Amrita means that is the like of the divine nectar, and the use of the Amrita, it is the Giloy, it is the Rasayana, and it is the Guduchi, Giloy, and there are a important name is the Amrita. It is used as a best immunomodulator, and I think the preparations, the Guduchi Kwath, Giloy Sattva and Giloy Dhanvati, it is already in the market. And this is the Giloy Sattva, the useful parts is the stem, and from the stem their extract as a powder, that is the known as the Giloy Sattva, and it is the important medicinal plant, and in the Jharkhand it is also known as Gulanch, and if it growing in the neem, it has some specific kind of activity because this plant is a climber, and if it grow in association of a trees like Arjuna, Jamun or Neem, it has some unique kind of efficacy also. This plant is useful for the diabetic, this plant is antipyretic, this plant is already mentioned and proven as a anti-cancer also. So at the end, it has been proven as a immunomodulator medicinal plant. Some years ago, there was a problem of the dengue, and this Giloy was used as a immunomodulator for the dengue affected people. So now in the COVID-19, people are using in the home remedies or as a home. If somebody are interested, they may take 1 to 2 inch, maximum 1 inch to 2 inch cuttings of the stem, and they have to they may crush, and they may take either aqueous extract or the decoction, but not more than 1 teaspoon, because more use of the Giloy, it may be constipation. And somebody is saying that some medicinal plant may be toxic. It may be, but that depends on the doses, what dosage you are using. If you are using randomly in a large quantity, it will be wrong. You have to take specifically as a medicine, because these are not a edible plant. It is a medicinal plant. It is not for the cooking and taking as a independently in the large quantity. So giloy, it is a best immunomodulator, and it is the very much utilizing by the people today. And through the Birsa Agricultural University, through the All India Coordinated Research Project on Medicinal and Aromatic Plant, governing by the ICAR and Directorate of Medicinal and Aromatic Plant. This is a mandatory crop, and people who are interested about to understand about the giloy, we are invited for the Birsa Agriculture University to see in our field. But after the lockdown, opening of the unlock conditions, and they may say, and it may grow in the home also, it may grow anywhere. So there is no more cultivation package is required, but it should to be always be organic. You can grow in the pot, you can grow in your home garden, and you can grow in your household premises, and you can cultivate in the field for the commercial purpose. And there are in the commercial purpose, they have to maintain some quality use of the industry. But the Guduchi Koat, Giloy Sat, Giloy Ganbati, it is the produce, and persons suffering from the infectious diseases, it is the prescription of the generally Ayurvedic physicians as immunomodulator. And people believe that this is very good plants for the management of COVID-19. This is a sampling view of the giloy. The stem are useful, and 1 year, after the 1 year to 18 months, mostly after the 18 month, it may used for the any kind of harvesting purpose. But in a twig, it should not to be utilized. It should to be always wait for the up to the minimum after the 18 months for their cultivation. Suppose somebody has urgent need to these days, they have to collect from the certain places where it is growing. It is also sometimes sold in the market, and there are some preparations which are in the Google or any kind of literature, it may be search out that this is giloy is the one of the important medicinal plant as a immunomodulator. Now the second plant is ashwagandha. Ashwagandha, what is the meaning? People are showing ashwa means ghoda, the horse. Ashwa means ghoda, horse, and gandha. It is the gandha of the horse. Ghode ka gandh? No. In the Sanskrit, it is the ashwagandhan mandheya iti ashwagandha. And घोड़े की तरह जिससे शक्ति प्राप्त हो जाए वो अश्वगंधा है. Giloy was propagated by the stem cutting, but this ashwagandha is propagated by the seeds. Because I am suggesting or writing here about the propagation, which are mostly depends for the commercial cultivation. Any kind of medicinal plant may grow through the seeds, no doubt. But if you are going for the large scale of commercial cultivation, so it is very much important. It is very much issues to how you recommend for the cultivation in a commercial cultivation. This is a photographs of our Birsa Agriculture University for the cultivation of ashwagandha. So ashwagandha, now there are several kind of agency like ICR, CSIR now discussing about the promotion of cultivation of ashwagandha in perspective of COVID-19 management. This is one of the important medicinal plant for the potency. Ashwagandha is ashwagandhadi bati, and there are several kind of preparations and produce are used for the manufacturing of herbal and Ayurvedic product. So ashwagandha is one of the potential medicinal plant for the management of COVID-19 as a immunomodulator. This is the preparation, and this is the roots are, as giloy was, the stem was useful, but its roots have high potency and tonic. The Ashwagandha roots and Ashwagandha, the virenin, somniferin, in some secondary metabolites are found to be the active principle, active constituent, which are found very good for the health. Now, one of the plant Biotech Nirgundi, in the Jharkhand, it is called Sindhuar. And the Nirgundi, it is the Nirgundi Shariram Rakshati, जो शरीर की सब प्रकार से रक्षा करता हो, वही निर्गुंडी है. It is a health protective, and it is propagated by the stem cutting, and the stem barks and leaves are very much anti-inflammatory, rheumatism, but it is very useful. The Nirgundi Kalp, Nirgundi Tel, useful for the cough, fever and body ache. People in the COVID-19 are generally suffering. Their complications are the respiratory diseases. Their respiratory tracts are being infected, and it is through the nose, through the breathing. There are certain kind of inhaling conditions. They get infection, and ultimately the symptom is the cough, fever, body ache. And Nirgundi has been mentioned that the Nirgundi Kalp, Nirgundi Tel, the preparation of Ayurveda is useful for the cough, fever and body ache. And in the local health tradition, Biotech Niganto is potentially utilized for the treatment of such kind of treatment and complications. Now, one of the important plant is the Vasa, Edatoda gelenica. Vasa कहते हैं कि जैसे कि कोई खांसा, मतलब जैसे ही कोई खांसने लगा, जैसे कोई खांसा तो दो उसको खिला के वासा. It is in local health tradition. In the COVID-19, the respiratory tract are being infected. And Eratoda gelenica, the Vasa, is a very good important plant. And we can say in the Jharkhand or in the Gangetic Plain, in the Deccan Plateau, in the Chotanagpur Plateau, and up to the Himalayan foothill area, Vasa is all where is occurring. And the extract of the leaf of the Vasa, it is very much useful for the cough or respiratory tract diseases. Problem is that we are just ignoring our local health tradition. We are just not providing the weightage to some medicinal plant which provided by the nature. Because nobody can beat the nature. कोई प्रकृति को beat नहीं कर सकता. And nobody can prepare such kind of medicinal plants which are given by the nature. So the Vasa is very much the Vasa Vale, Vasa Risht, Vasa Panak in the Ayurveda. And in the folklore, in the travel medicine, in the ethnomedicine, the extract of leaves is used for the relief from the bronchitis or cough or respiratory diseases. And as you know, in the COVID-19, people are suffering from such kind of problems. So the use of Vasa may be useful. And it is considered as immunomodulator plant, because now the COVID problem is the whole kind of arrest the respiratory tract infections. So the Vasa is a very good medicinal plant. Now, one of the medicinal plant is the Pipali. Pipali, Piper longum, and the fruits used for cough and respiratory diseases. It is propagated by the seeds. Some people are interested for the cultivation. They may propagate through the seed. It is the Piper longum, and the Tricotu is also prepared through the help of the adrak, ginger, and this Piper longum. It is a very good combination, and in addition, people are interested, they make some Piper nigrum also, Kali Mirch. So these have been found very good effective for the cough and respiratory diseases treatment. It is propagated by the seeds. Now, it is a medicinal plant, Kalmeg. Everybody knows in the Jharkhand, especially in the Deccan Plateau, and it is one of the important plant which are, somebody are saying this is Chirata, but it is not Chirata. Chirata is Suercia chirata occurring in the Himalayan region. And this is the Kalmeg. And it is Kirata, Ayurvedic Kirata. It is found everywhere in the Jharkhand. It may be cultivated very easily. And it is one of the important medicinal plant for the blood purifier, antipyretic, cure liver disorders. And it is propagated by the seeds. So the Kalmeg, don't confuse about this. This is the Chirayata. This is never been Chirayata. It is the Kirayata. And it is already documented in the Ayurveda, it is Kiratatik. And where Chirayata is not available, it is just utilized as an alternate. Kalmeg is utilized, but it is a very good. It is a very good medicinal plant for the blood purifier, antipyretic, cure liver disorder, and there are certain modern herbal modern products where the liver tonic, like you just purchased the medicine Trisoli. There is neo-andrographolide and andrographolide contents, the active principle are in the medicine. So isolated compound when utilized, it is just used as a modern medicine. But in the folklore, Kalmeg is very much useful as a immunomodulator for certain kind of complications. And now aloe vera, a highly commercial medicinal plant, and it is the one of the important medicinal plant. Large number of herbal products manufactured from the leaves of the plant, useful for hand sanitizer. Now there are many herbal products where the pulp of the aloe vera is used for the hand sanitizer. It is the cosmetic products. It is the skin care. It is the juice is used for the liver disorders. And if you go through the search of the literature, it is aloe vera or Gharif Kumari, it one of the leading medicinal plant in the world for the commercial importance, because most of the beauty products for the cosmetic products are manufactured. And in the COVID-19 era, these leaves pulp are used as a sanitizer. We have developed in the Ranchi district aloe vera village in the Nagri block, that is the Devdi village, which we have referred for the aloe vera village, and there is a good selling of the aloe vera medicinal plant. And through the Birsa Agriculture University, we are developing as a independently. So the aloe vera, the leaves are very good, very useful for the hand sanitizer, and with the isopropanol or alcoholic solution, it is just to mix up and utilize, because somebody have suffering the problem to wash always through the soap. And soap is very good for the washing or sanitizing their hands. But regular washing of your hands, it may be that some problem may created through the regular use of the water and water cleaning the hand. So if the hand sanitizer with the help of aloe vera, so it is the one of the important plant. Next. It is a view of the aloe vera cultivation of our Birsa Agriculture University, and people are saying that we are just growing, and the pulp, this is the method of the pulp extraction. because covering all the processing. So this is the method of the pulp. There is a pulverizer. When the cuttings, their core of the cuttings of the leaves, it is used for the extraction of. This is the instrument we are in Birsa Agriculture University for making the pulp extraction. Next. This is the another backside view of the aloe vera, and it is the homogenizer, the remove pigments, and this through the homogenizer, the pigments and latex which are in the leaves is to be removed, and through the homogenizer, the preservative for the storage condition of at least 6 months. There are some permissible preservative are used, but all process are being done in the homogenizer for the stabilization of the juice, aloe vera juice. One of the important plant is the Punarnava. It is the khapda saag, and in the Jharkhand it is highly used by the ethnic people, and it is very good use for the rejuvenating, hepatic care, urinary trouble, and Punarnava Punarnavinam Karoti, जो बार-बार शरीर को फिर से नया कर सकता है. That is the Punarnava, Boerevia diffusa, family Nictagenaceae, and Punarnavadi Mandur and Punarnava Astak. It is very good preparations, herbal preparation. It is immunomodulator and utilized, and in the particularly Santhal people in the Santhal Pragna, they are cooked as vegetable of this and just taking as a vegetable in their food systems. So, and in the Ayurveda, it is Punarnava, it is very good, important medicinal plant as a immunomodulator. Now, one of the medicinal plant is Tulsi. कहते हैं कि तुलांग अस्यतीति नास्यतीति, जिससे किसी की तुलना नहीं कर सकते हैं, वही तो तुलसी है. जिससे किसी की तुलना, it is the holy tulsi. The name is Ocimum tenuiflorum, but the Ocimum sanctum, it was the earlier name. Now it is Ocimum tenuiflorum, family Lamiaceae. The leaves useful for the cough, antipyretic, and it is propagated by seeds. And the market, the tulsi earth is selling leaf decoction, leaf extract, may be used as a home remedies, and there are organic tea, tulsi based tea. It is already in the market. There are a very good market of the tulsi seed oil, the tulsi leaf oil, tulsi arc, tulsi powder. It is all are used. And if somebody is going to use tulsi leaf or whole plant of the tulsi or the extract of the tulsi, leaf decoction of the tulsi, extract of the tulsi, and putting some 3 or 4 at least leaves in their daily for the using as a home remedies, it is assumed that they may get relief from the cough, antipyretic. And there are not side effects have been reported, because side effect is of the like modern medicine, it is very complicated problem. But in the Ayurveda, there is not certain kind of toxic side effect. If there is some kind of constipation and some kind of very easy kind of problems may create, but not kind of acute. So it may be used as a home remedy. So Tulsi, it is the highly important medicinal plant. Now processing is a very versatile tools of value addition. And processing, because if somebody is saying, okay, I have developed or cultivated very good medicinal plant, but it is not processed properly within the time frame, within a particular quality control condition, scientifically, so it all be useless. If some plant collected, it should to be next. The general procedure of the processing is post-harvest technology adoptions, because somebody has cultivated but not properly harvested, and in a particular season, in a particular period, in a particular controlled conditions, if it is not harvested, then the whole work will be deteriorate. Say dry of the useful plant, it should not to be dry in the direct sunlight, because the most of the important ingredients or active principles, scientific principles may be vanish. Through oven drying at optimum temperature, utilization of tools or equipment scientifically improved, it should to be followed. Following WHO guideline of GSEB, good agriculture and collection practices, good manufacturing practices, maintain pharmacopoeia standards, parameters, quality controlled, available in government official websites. I use, if you go to search the what will be the parameter, you go search in the Google about the Ayurvedic pharmacopoeia, you will just obtain the pharmacopoeia standard. It is official publication and it should to be mentioned. So persons who are interested to cultivate for the marketing or medicinal plant, they have to see the rules of the general rules, because it will be utilized for the pharmaceutical industry for the quality control. Now, as I have discussed, some plants like giloy, ashwagandha, nirgundi, vasa, punarnava, tulsi, it is due to there are a certain limit of times which I have to speak here. These are very good medicinal plants, and it is already reported. These are used as some plants like giloy is used as a rasayan, but like use of the amla, it is the source of vitamin C. It may be used. Some fruits may be taken. There is some kind of a large number of fruits are important for the use as a daily for the daily consumptions. But some medicinal plant exclusively may be helpful to, because in the COVID-19 problem, the cough and such kind of disorders for breathing problem is started and infected. So the basa is very much useful. Tulsi is very much useful. The ashwagandha, which is particularly useful for the potency, giloy is immunomodulator, it reduce some kind of fever condition. So there is need for large scale cultivation and processing of medicinal plant to ensure availability of good quality planting materials. The natural medicinal plants has high valued in perspectives of COVID-19 as immunomodulator. Second thing, I do not want to say any kind of claim that this is the only medicinal plant may be useful, and this is one of the medicinal plants aspects which are useful for the management through the immunomodulator. We are not going to claim any kind of claim. So this is the important medicinal plant already mentioned in local health tradition of the Ayurveda, and it is also used as a ethnomedicine, and it is used as a home remedies or folk remedies. So these plants may be utilized for the as a immunomodulator, as a health protective, for the therapeutic, curative, and infectious properties.
Thank you very much, sir. Thank you for your excellent presentation. Now we have questions from the participants. So kindly answer to their queries.
Yeah.
Sir, the first question is, what would be the quality parameters of raw material of cultivated immunomodulator medicinal plants? Quality parameters.
Yes, it is a very good questions. I appreciate, because without quality, there is no value of the cultivated produce. Because if you want to interested to sell in the Ayurvedic pharmacies, they have to see the Ayurvedic pharmacopoeia standard. Suppose some plants, some persons have cultivated the ashwagandha root. They have to go to the website. What is the quality control parameter? After the shade drying, what will be the percentage? what will be the alcohol soluble extractive, what is the criteria of their powder. There is certain kind of fluorescence analysis. If somebody are selling in the powder form, there is another quality parameter. It should to be evaluated under the UV light. Some person interested to sell in the crude form, like they are interested to sell the root. So if some are selling the root, there is a morphological characteristics. Some are interested to sold in the some crust form. So there is important to understand about the anatomical or histological studies. So it all has mentioned in the Ayurvedic pharmacopoeia. They have to refer, and there are certain books published for the cultivation of medicinal plant. There is also mentioned, but the best is to refer. They have to see the Ayurvedic pharmacopoeia for the quality control parameter. Because it is the standardization manual. It is already published. Even ICMR, Indian Council of Medical Research, has also published the Ayurvedic Pharmacopoeia, Indian Council of ICMR publication. It all covers about the standard, and Ayurvedic pharmacies are regularly following this.
Thank you, sir. Another question is from Parul Banerjee. She asked, I read that in several cases, it is the immune response of the body which kills the patient. Therefore, the immune response may be more dangerous than virus itself. Is there a herb that may be helpful in modulating the amount of immune response?
Yes, it is a good question also. Immunomodulator means you have to, there is some develop of the protective body himself prepare for the protective. Now, as I have told that the uses of, I am talking presently about the situations, because suppose I have discussed about the giloy. It is very good immunomodulator. I have already described it. तुलसी, वासा, अश्वगंधा, अश्वगंधन मानधेय इति अश्वगंधा, जिससे घोड़े की तरह शक्ति प्राप्त होती है कहते हैं. It is the अश्वगंधा रिष्ट और अश्वगंधाति बटी. It is a very good potency. Suppose you will see what kind of immunomodulation we will achieve. If you will achieve the rog pratirodhak क्षमता, so resistance in the body, you can fight any of the infectious diseases. So there is no problem. So giloy, punarnava, ashwagandha, vasa, tulsi, such kind of medicinal plants may be utilized, but the utilization pattern should be also to be followed as per the guideline, not randomly, not just to in the huge quantity, because somebody will say that I will go to the use a large number of giloy kadha. It will be constipation will be occur, because somebody will be use more tulsi leaf. It is possible that it contains essential oil. So in the limit quantity, if people are utilizing such kind of medicinal plant, it will be very good for the management of as a immunomodulator. There is no toxic in a specific parameter, and as a home remedies, people are utilizing still. And for the exclusively for if you want to earn the money and through the marketing, this is one of the important tools and techniques which I have explained for the commercial cultivation.
Thank you, sir. Next question is, does processed and packaged plants differ from fresh plant product, and to which extent?
Yes, it is very good, important question. I thank to the who is asking, because suppose we all are interested to obtain medicinal plants exclusively. Suppose some medicinal plant growing in the garden, and you have required this medicinal plant, you cannot use fresh. So the crude raw material is required for the availability of general public. There are certain medicinal plant growing in the Himalayan region. Somebody is interested to use the medicinal plant as a fresh. It is possible. There are some medicinal plant growing in the Himalaya like Kutki Kut, Barberries, Daru Haridra. There are a certain medicinal plants which are occurring in the Rajasthan, like Google. It is how we can use phrase. So for the availability, there is a control parameter conditions which has been recommended for as a quality parameter. But still, somebody is saying that the fresh leaf of tulsi, it is obviously it is efficacious. And but for the pharmacies, for the large number of manufacturing of the products, there is a method of pharmacy scientifically to harvest their quality like fresh. But some plants which are used freshly may be efficacious, no doubt. But for the manufacturing of large number of products, there is need of the maintain of the quality parameter. But fresh always not be available as a home remedy. Somebody has taken 2 or 4 leaves or 5 leaves. But in a long term, we have to do the practice of the commercial cultivation through the post-harvest technology, through the drying, and for the availability to everyone.
Thank you, sir. Another question is, what is the marketing potential of tulsi? This is the last one.
No, you can ask any question. I have enough time because I was very much interested to attend. If you have no time, I have no problem, but I have full time. The tulsi leaves have now the tulsi leaf is one of the important medicinal plants widely selling in the market. The organic tea. Organic tea is prepared by the tulsi leaves, and as a deep tea, people have avoided manywhere for the use of the tea. They are using tulsi tea. It is just as used for the cough. It is useful for the fever. It is immunomodulator. You will feel fresh. Disease will be, and if you go the search of their pharmacological efficacy of the tulsi, madam, you cannot imagine. There is a large number of potential usage of tulsi has been described. It is another thing that it is a religious plant or spiritual plants, but tulsi has still a very potency. And tulsi arc, as I have told, there is a great market. The tulsi tea, like a organic tea, and the tulsi bati is also being prepared, and there is in the market tulsi. So there are certain products of tulsi which are osimum 2 or 3 or 4 or 5 osimum species, and which are utilized tulsi. The whole extract are as a drop. It is also sold in the market. I do not want to tell about the products due to some reason, not for the promotion of the marketing, but there are certain kind of herbal products which juice are used for the as a extract, as a tulsi drop in the market, and people are utilizing. So there is a great and large demand of the tulsi, but it should all very much organic, because if there is contamination through the environmental, through the toxicological, so it will be certain problem. So the maintenance of is important.
Thank you very much, sir. I would like to ask Dr. Indu to propose vote of thanks.
On behalf of Nirmala College, I thank our guest, Dr. Kasil Kumar, for his knowledge, giving untiring speech here in 2-day national webinar. We understood significance of immunomodulator plants like giloy, ashwagandha, tulsi, kalmik, and other medicinal plants. Cultivation and processing of industrial purpose related to herbal pharmacy, and regular supply of good quality of raw material for manufacturing of herbal products. Thank you, sir. Thank you, everyone, for helping to organize such a wonderful session successfully. Thank you.
Thank you, Dr. Indu.
I am also thankful to all the organizers.
Thank you.
I am also thankful to all the organization who invited me for deliver this lecture. So I am thankful.
Thank you, sir. Sir, we are leading towards closing ceremony. It is the time for closing ceremony of this 2-day national webinar. We have with us our patron, Dr. Sister Jyoti, respected patron Dr. Sister Jyoti, Principal, Nirmala College, Ranchi, respected resource person, Dr. Kasil Kumar, Vice Principal, Sister Shobha, co-convener, Dr. Anubhuti Singh, and my dear participants, warm greetings to you all. I'm sure this webinar was a fruitful one. You are taking lots of ideas and knowledge how to protect yourselves from COVID-19, which is going to have a long-lasting scar on our lives, whether it is health, policy, economy, everywhere, in every sphere of life, in every country, it is going to have some and other effect. Now, I'm sure that this knowledge of pandemic COVID-19, how we can cure it or how we can save ourselves is going to help us a lot. I request co-convener Dr. Anubhuti Singh, Assistant Professor, Botany Department, Nirmala College, Ranchi, to present the report of this webinar. Over to Dr. Anubhuti.
Thank you, Dr. Emma. Honorable Principal and Patron Dr. Sister Jyoti, Vice Principal Sister Shobha, eminent speakers, Convener Dr. Emma Seraphim, members of Organizing Committee and all the participants. Greetings to all. On behalf of Organizing Committee, I, Dr. Anubhuti Singh, honored to present the report on successful 2-day national webinar on topic pandemic COVID-19, pathophysiology, prevention and treatment. Our webinar witnessed 300 participants across the country from MP, UP, Bihar, Karnataka, Tamil Nadu and other states. Altogether, 6 sessions took place. The first session on 10 August was on the topic Source, Transmission and Pathogenicity of Coronavirus by Professor Anilava Kaviraj, Department of Zoology, University of Kalyani, West Bengal. The second session was on topic Diagnosis, Prevention and the Treatment of COVID by Dr. Ashok Kumar Sarma, Associate Professor, Department of Microbiology, RIMS, Ranchi. And the third session of the day, one was on role of immunity booster food in the wake up of pandemic by Dr. Sudarshan Maurya, Senior Scientist, ICAR, Varanasi. On 11 August, we had Professor Partha P. Mazumdar, Professor and Founder, National Institute of Biomedical Genomics, Kalyani University. He talked on the topic Evolution and Spread of Sars Cov-2. In the second session, we had Dr. Kakali Bhadra, Assistant Professor, Kalyani University. She talked about synthetic drugs to natural metabolites, a retrospective. Our concluding session was shared by Dr. Kasil Kumar, Head of Forest Products and Utilization, Birsa Agriculture University, Ranchi. He spoke about cultivation and processing of immunomodulator medicinal plants. Thus, our national webinar has altogether 6 sessions covering all the aspects of COVID-19 from its source, transmission, diagnosis, its evolution, and to its prevention through the immunomodulator medicinal plants. I would like to express my gratitude to our principal and patron for providing us continuous support, guidance, and encouragement. Thank you, sister, and thank you all.
Thank you, Dr. Anubhuti. Our beloved patron and principal, Dr. Sister Jyoti, she is the inspiration behind this event, this national webinar. It is her motivation and guiding spirit as well as cooperation that keeps us going. Recently, she has got Corona Karamveer Samman under Rashtra Srijan Abhiyan. And we once again congratulate her on behalf of all who are present here. Congratulations, sister. With immense love and gratitude, I humbly request our patron and beloved principal, Dr. Sister Jyoti, to deliver the closing ceremony address. Over to sister.
Thank you, Dr. Emarani. Wish you all a very warm and blessed good afternoon.
Sister.
Our esteemed guest and resource person of the final session of the two-day national webinar, Dr. Kasil Kumar, Birsa Agriculture University, Ranchi, organizers Dr. Emarani Seraphim and Dr. Anubhuti Singh and their entire team, faculty members, distinguished guests, and dear students, all participants from far and wide. Someone has rightly said that thinking is progress, non-thinking is stagnation of the individual, organization, and the country. Thinking leads us to action. Knowledge without action is useless and irrelevant. Knowledge with action converts adversity into prosperity. Indeed, this is what I have to say to the organizers today, Dr. Emarani Seraphim and Dr. Anubhuti Singh and their entire team. First of all, I thank the Almighty for giving us these two grace-filled days with numerous blessings. I feel proud to say that the two-day national webinar was full of relevant information related to COVID-19. My heartiest congratulations to the organizing departments of Botany and Zoology. I myself believe that there can be no limit to achieve excellence if we really have the willpower to do so. The world has witnessed moments of happiness as well as despair. But as we know, nothing is forever. So one day, surely we would find ourselves free of this vulnerable infection that has created a standstill. Undoubtedly, there will be scars of its devastation, but we need to cover it up through our efforts to evolve methods of precaution and safety. If every one of us plays our own role of safety, it's definite to fade its severity. Obstacles can slow down our progress, but it cannot stop us from excelling. It's important to understand that the only way to escape this pandemic is to face it by using the best precautionary measures. This webinar has undoubtedly added to our understanding of the problem and will help us to stay protected and make others remain protected. This is surely the best way to serve humanity. Hopefully, we shall soon breathe in a freshness of corona-free world where keeping a mask, social distancing, and lockdown would not remain inevitable part of our vocabulary. I hope and pray for a beautiful world with the best virtues. I'm sure that all the speakers have contributed their knowledge bank with us to enhance our knowledge and enrich us. I express my heartfelt thanks to all speakers for giving positive response to our invitation and become part of Nirmala College, Ranchi, affiliated to Ranchi University, Ranchi, a college with potential for excellence, accredited by NAC with grade A. Here I place on my heartiest congratulations to the organizers for their commendable efforts and commitment. A big thanks And congratulations to all the participants for being part of this great event. May God bless you all. Thank you.
Thank you, sister. Thank you very much, sister, for this beautiful, encouraging words to us. Now, may I request Dr. Anjali Smita, Assistant Professor, Nirmala College, Ranchi, to deliver the vote of thanks.
Thank you, ma'am. On behalf of the Department of Botany and Zoology, myself, Dr. Anjali Smita, Assistant Professor, Department of Zoology, extend my gratitude to our patron and respected principal, Dr. Sister Jyoti, for her unconditional support, who motivates us in every sphere of our life. I'm thankful to our Vice Principal, Sister Shobha, for her relentless help. I am extremely thankful to the eminent resource person, cum speakers, Dr. Anilav Kaviraj, Dr. Ashok Sharma, Dr. Sudarshan Maurya, Professor Parth Majumdar, Dr. Kakali Bhatra, Dr. Kasil Kumar, for enlightening us with their exotic presentations. My sincerest thanks to the convener, Dr. Emma Seraphim, HOD, Department of Zoology, and co-convener, Dr. Anubhuti Singh, Department of Botany, respectively, for taking the pain to organize this wonderful event. My heartfelt thanks to members of organizing committee, Dr. Veena Pani, Dr. Manisha Kumari, Dr. Indu Kumari, and Dr. Neetu Rani for their ceaseless efforts. I'm grateful to all our participants that include my seniors as well as my colleagues to be with us and encourage us in the webinar. I extend my thanks to Mr. Aman Kumar Jha for helping us so well with his technical expertise. Once more, I thank each and everyone who directly or indirectly had been a part of this event. I pray that the Almighty bless us with more of strength to go for such academic gatherings. Thanks everyone and have a good day.
Thank you, Dr. Anjali Smita. May I request our dignitaries and participants to arise for national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तव शुभ नामे जपे तव शुभ आशीष मांगे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे जय जय
Thank you one and all. Stay safe from coronavirus. Stay safe and stay healthy. Thank you once again.